Looks like we've started. Good morning, everyone. Thank you all for joining us today. Today's event is hosted by the Office of Therapeutic Products, or OTP, in the Center for Biologics Evaluation and Research at the U.S. Food and Drug Administration. My name is Mark Verdesia, and I am a biological reviewer in OTP's Office of Gene Therapy, and I will be one of your moderators for today's event. I'm pleased to welcome. The objective of today's meeting is to hear from cell and gene therapy and application review. We look forward to hearing perspectives in CMC. We appreciate all of you joining us today for your thoughts on this important topic, as well as to everyone who's tuning in just to listen. Next slide, please. Great. As you can see, we have a full agenda that includes 3 sessions organized by product area. Before we jump into any of our sessions, we will first hear opening remarks from Acting OTP Director, Dr. Vijay Kumar. After Dr. Kumar's remarks, I will then provide an overview of the meeting process before we get started. Another point I want to make today is that during today's meeting, we will have some of my colleagues from FDA listening in and taking notes. At the end of each session, there will be some time for them to ask questions of our public speakers in order to follow up or clarify on anything. So if you are a public speaker, please stay on logged in for the entire session in case one of our staff members have a question for you at the end. Next slide, please. Great. So before we get started, I just want to go over some basic housekeeping items. First, the meeting will be recorded and is available and will be available online in the coming weeks. Closed captioning for this event is available directly in Teams. And we also ask that if you use the chat box to only use it to report any technical difficulties. During this event, all microphones for the general audience will be muted. Because this is a listening meeting, we will not have a Q&A portion for the audience, but we do appreciate your comments, and we encourage you to go to the docket at regulations.gov if you would like to share any specific feedback on anything discussed today. The docket number is FDA-2025-N-2218, and it will remain open until October 17th, 2025. All the comments in the docket, as well as the presentation from today's meeting, will be used to inform the writing of a draft FDA guidance document on the topics that we discussed today. Okay, and with that, let's move to the next slide, and I'll pass it to Dr. Vijay Kumar for opening remarks. Dr. Kumar, the mic is yours.
Thank you, Dr. Mark Verdesia. Good morning and welcome to this important public listening meeting hosted by the FDA and the Center for Biologics Evaluation and Research, Office of Therapeutic Products. My name is Dr. Vijay Kumar, and as Dr. Verdesia mentioned, I'm the Acting Director of OTP. I am honored to be here today to address this gathering of nearly 1,000 participants representing many of our valued stakeholders, clinicians, scientists, researchers, and patient advocates. Next slide, please. The cell and gene therapy field has experienced tremendous growth and transformation over the past decades. We have witnessed approvals that have fundamentally changed treatment paradigms for previously incurable diseases. And we have a broad range of products from CAR-Ts revolutionizing cancer treatment to gene editing-based treatments addressing rare genetic disorders. We continue to see promising advancements in the realm of cell and gene therapy. Currently, there are 46 approved cell and gene therapies and over 3,600 active INDs in our office, which is remarkable considering there were only a handful of cell and gene therapies approved only a decade ago. Next slide, please. Despite this tremendous progress, the field faces several persistent challenges that require collaborative solutions. For example, manufacturing complexity and cost still remain a significant hurdle, with many cell and gene therapy products requiring specialized facilities, sophisticated quality systems, and highly trained personnel. Reducing and or replacing the use of animals. Heterogeneity within the patients with rare diseases and even within cell and gene therapeutics themselves means that the traditional regulatory approaches must evolve to accommodate innovation while ensuring patient safety. Unlike traditional pharmaceuticals, where established precedents can guide development, many cell and gene therapy products are first in class with limited regulatory history. This novelty, while exciting, can create uncertainty for both sponsors and regulators. Additionally, the long-term safety profile of many cell and gene therapy products require extended follow-up periods, creating challenges in study design and regulatory decision-making. Striking the appropriate balance between the urgent need for breakthrough therapies with comprehensive safety evaluation remains a challenge. The regulatory landscape has adapted significantly to support cell and gene therapy innovation. We have developed expedited pathways, implemented risk-based approaches to help advance product development for rare diseases, and enhanced our scientific expertise to keep pace with technological advancement. Yet we cannot rest on our laurels. We recognize that continued evolution is necessary to meet the growing pipeline of cell and gene therapy products seeking regulatory approval. Next slide, please. The primary purpose of today's meeting is to hear your perspectives about how we can leverage prior knowledge and experience to facilitate both product development and regulatory review in the cell and gene therapy space. We are here to listen and learn about strategies that can streamline pathways while maintaining the highest standards of safety and efficacy. Specifically, FDA wishes to explore and gather insights in three critical areas. First, what types of data and information are sponsors willing to share that could advance cell and gene therapy product development and regulatory review? These insights may help us identify opportunities for more efficient development pathways. Second, how can data leveraging occur between external partners, including contract manufacturing organizations, contract development and manufacturing organizations, and their licensees across the full product cycle? Learning about how this type of collaboration is being managed now and the potential strategies for future collaboration may serve to streamline cell and gene therapy development. Third, how can data and information be effectively integrated across multiple disciplines to enhance development, manufacturing, and safety assessments of cell and gene therapy products? The complex nature of cell and gene therapy products and the diseases they intend to treat require interdisciplinary crosstalk to increase efficiency in development while ensuring patient needs and safety are being met. Today marks another significant milestone in our collective journey to advance cell and gene therapies. Your presence here demonstrates our shared commitment to bringing life-changing therapies to patients who need them the most. Next slide, please. This brings us to today's central theme. A part of our mission, as laid out on our website, states that FDA is responsible for advancing the public health by helping to speed innovations that make medical products more effective and safer. Today's meeting is one of the steps to operationalize that aspect of our mission, specifically how leveraging data and prior knowledge can accelerate the path from laboratory bench to the patient bedside. When we can thoughtfully apply lessons learned from previous development programs, we may avoid unnecessarily repeating studies and focus our precious resources on addressing novel aspects of each product. This type of data leveraging offers multiple benefits to different stakeholders. For sponsors, it can reduce development timelines, decrease cost, and minimize redundant studies. We also hope to listen about possibilities for how data can be leveraged between related indications or applicability towards different demographics. For regulators, leveraging can enable more informed decision-making based on broader data sets and accumulated experience. Most importantly, for our patients, it could mean faster access to potentially life-saving treatments. The key is developing frameworks that allow us to appropriately leverage from prior experience while we maintain rigorous scientific standards. This requires careful consideration of product similarities, patient populations, and manufacturing process to determine when prior knowledge can inform current decisions. Today's discussions will inform the development of a draft guidance on leveraging prior knowledge in cell and gene therapy development and review, a PDUFA 7 commitment. This guidance will provide clarity and consistency for sponsors while supporting FDA's review process. The perspectives shared today will be instrumental in shaping this guidance. We encourage you to think boldly about possibilities while remaining grounded in scientific rigor. Consider not just what type of data sharing is possible, but what mechanisms could facilitate this sharing while protecting proprietary interests and competitive advantages. In addition to this meeting, as Dr. Mark Verdesia mentioned, FDA has also opened the docket and is accepting comments through October 17, 2025. We encourage all our stakeholders to share their perspectives and ideas through the docket, as those comments will also be used to inform the draft guidance. As we begin today's sessions, I want to emphasize that this meeting represents more than just a PDUFA commitment. It's an opportunity for all of us to collectively advance a field that holds tremendous promise for patients worldwide. The ideas shared today can help accelerate the next generation of breakthrough therapies. At this time, I would like to thank you for your participation, your expertise, and your commitment to help advance cell and gene therapies. We look forward to hearing your presentation and remarks. And with that, I will pass it on to Dr. Verdesia to continue with today's public listening meeting.
Thank you, Dr. Kumar, for those opening remarks. So now I'd like to give a brief overview of our meeting process, which should be fairly straightforward. The moderator of each session, and that's me for session 1, will introduce the speaker so that they can begin their presentation. When it is your turn to speak, you can unmute yourself and start your presentation. Once your presentation is done, we ask that you go back on mute and allow the next speaker to present. Our first group of speakers in session 1 should already see microphone and camera access. If you are a session 1 speaker and you do not have microphone or camera access, please let us know in the chat box. For speakers in session 2 and 3, we will grant you access to the camera and microphone as we get closer to your session time. Okay, for those of you who submitted slides, your slide presentation has been added to this slide deck. When speaking, please just let us know when to advance the slides. Also, To ensure transparency, we encourage you to advise the audience of any financial relationships that you may have with any firm, group, company, or product at the start of your presentation. If you do not have any financial relationship as such, you can simply make a statement to that effect. Please keep the time limit in mind when speaking. Each speaker will have 5 minutes, and if you run out of time, we encourage you to submit any remaining comments to the docket. We do have 30 speakers today, and so it's really important that we try to stick to the time limits for each presentation. Next slide, please. As I mentioned a few minutes ago, we have some FDA staff who are listening in and taking notes during today's meeting. I'd like to take a moment now to introduce the FDA OTP subject matter experts who are serving on today's panel for session 1. First, we have Dr. Young-Wook Choi, Bioinformatics Reviewer in the Office of Cellular Therapy and Human Tissue. We also have Dr. Stella Lee, CMC Reviewer in the Office of Gene Therapy. And we also have Dr. Katarina Tislu, Medical Officer in the Office of Clinical Evaluation. And we have Dr. Emily Wires, PharmTox Reviewer in the Office of Pharmacology Toxicology. Thank you again to our FDA panelists for your time today. Next slide, please. Okay, great. We can move right into session 1. We have 13 speakers for this session. The topic for this session is gene therapy products, excluding those that utilize gene editing. The gene editing products will be discussed in session 2. I will be introducing each speaker right before I give them the floor, but I want to remind the speakers to restate their name and affiliation and to let us know about any financial relationship, if any, that you may have at the start of your presentation. Okay, so we're ready to start. Our first speaker for session 1 is Mercy Al-Ai, speaking on behalf of BIO's Regenerative Medicine Committee. Whenever you're ready.
Thank you. Thank you very much for the opportunity to present. My name is Mercy Al-Ai. I'm with Kite Pharma, which is a Gilead company. However, today I'm presenting on behalf of Bioregenerative Medicine. I just wanted to mention that the opinion and topics that I will be presenting are collection of our work through the Regenerative Medicine Committee and not necessarily my opinion or my company's position. If you could go to the first slide, please. Yes, thank you. Maybe next. Okay. We believe leveraging prior knowledge presents a huge opportunity for us. It's currently difficult to understand how to apply prior knowledge due to lack of clarity and acceptance of data that's not application specific. This duplication and repetition of studies definitely take additional time and delay the development of the drugs that are much needed for the patients. And by having this opportunity, we could make United States a more attractive place for drug developers. We propose that prior knowledge to be applicable in every phase of development. It could include pre- and post-market settings, and it would facilitate development and making process improvements, enhancements, and ultimately serve more patients and make products available to this patient population. Next. We think prior knowledge can come from many different sources and not limited to approved products. We believe many sources of these prior knowledge could be approved BLAs, also marketing authorizations. It could be INDs or CTs, clinical trial applications. It could come from collaborative or consortium efforts, and obviously scientific literature. And this prior data definitely can be used with the greatest impact in the areas of rare, ultra-rare diseases where there's unmet medical need and for the priority programs. Next. I am a CMC person, so what I'm going to focus is a lot of CMC examples on that we could use the prior knowledge, but it's definitely not limited to CMC and it could go across other disciplines. There are some examples of where prior knowledge could be used, for example, when you're making process improvements, analytical methods, leveraging methods from one product to another, viral clearance studies, stability data, setting shelf life for your product, specification setting, and manufacturing site qualification inspection. This is not an exhaustive list, but just some examples. And we like to propose that prior knowledge may be applicable across the field and could be product agnostic. There are examples of standardized methods such as transgene expression or potency for gene therapies. Next. This is my last slide, but I think the most important one. This is our ask from the agency. We believe that the use of prior knowledge can be applicable to data from any phase of the study, whether it's pre or post-approval setting. And this data could be product class, assay agnostic. And it's on us to make the scientific justification and connection and present it to the agency. We therefore ask for FDA to consider a flexible risk-based framework for assessing prior knowledge. And it's on us to look at the risk, to look at the degree of change, level of potential impact on safety, efficacy, and any residual uncertainty based on totality of evidence. And this level of risk will tell us how much we can leverage that data or what additional data we need to generate in order to be able to comfortably make the case. At the end of my presentation, I just want to make sure that I touch on some immediate areas of need for data to be leveraged. We think we need to streamline and address OOS out of a specification with the use of prior knowledge. No matter what, in the area of gene-modified cell therapy or gene therapy, the OOS out of a specification causes delay of access to the patients and subjects. We need to urgently address this and allow to revisit specification based on collective knowledge and maybe what we learn from one product to be expanded to another. Potency assays and CQA developments are also very critical for immediate addressing and attention of the agency. If you could provide us with more guidance on how we could leverage knowledge in addressing these requirements. Thank you.
Great, thank you very much. Our next speaker is Kelly Brazzo on behalf of Cure LGM D2I Foundation. Whenever you're ready.
Hi, thank you. Yes, my name is Kelly Brazzo, and I am the co-founder and CEO of Cure LGM D2I Foundation. My only disclosures is that Cure LGM D2I is a minor shareholder in ML Bio, Myogenica, Kinea Bio, and Ventura Life Sciences. Next slide, please. My daughter Sammy was diagnosed at the age of 2 with LGMD2I R9. At that time, we were told there's no treatment, try to keep her healthy, but she'll likely lose her ability to walk in early adolescence. 15 years later, she's now 17, and although you see her smile, what's hiding behind that smile is pain, loss of independence, and the fear of her uncertain future. Next slide. The LGMDs have over 30 subtypes, each caused by a known genetic variant. Symptoms start with progressive weakness, starting in the hip and shoulder girdles, and many subtypes involve respiratory and cardiac complications with an eventual loss of ambulation. Currently, there are no approved therapies for any LGMDs. Next slide. We have made excellent progress due to the collaborative efforts of many patient advocacy organizations. Together, we've established an LGMD care guide, standards of care, natural history studies, and patient registries. We had a patient listening session and an ELPFDD, and more recently hosted a scientific workshop bringing together clinical and scientific experts, industry, advocates, and the FDA. We also provide patient conferences and educational webinars to engage our patient community. And of critical importance, we've established ICD-10 codes for the LGMDs, priming the community for much needed and awaited treatment approvals. Next slide. Sadly, over the last few months, there have been several gene therapy programs for the LGMDs that have been paused or stopped altogether, leaving many families lost and disheartened. Most subtypes are ideal candidates for gene therapy due to the small size of the affected genes. Each of the LGMDs are ultra rare and progressive, fit for the accelerated approval pathway or the recently announced RDEP program. However, the cost of developing treatments only one subtype at a time is unsustainable, and patients don't have time to wait for that process. Next slide. I'm here today with a plea for your support in accelerating drug development for our patients. We could utilize platform designation for the LGMDs that have a common protein dysfunction or where common target organ expression is needed. We could leverage assay validation and process development across programs that utilize common capsids. Next slide. Our community is asking for transparency on data collection, trial design, manufacturing, and any serious adverse events. Sharing of data also reduces duplication and ultimately can save time and muscle for our patients. Our advocates are willing and able to serve as neutral conveners between sponsors and regulators to ensure trial design that is meaningful and impactful. Patients should be given a seat at a table to develop and determine the risk-benefit frameworks for potential treatments. Next slide. Our registries and natural history studies provide critical baseline data across subtypes. We can utilize the patient voice to help define meaningful clinical endpoints and relevant patient-reported outcomes. For example, instead of just using the NSAID or the PUL for non-ambulatory patients, we could look at workable space improvement, like using a mouse or utensils for self-feeding. Traditional clinical endpoints are not realistic for many of our patients who already have significant progression. Patients have expressed that even if they can maintain their level of function, that would be considered a successful outcome. They just can't afford additional losses. We also ask that the data be shared, which could speed regulatory pathways and minimize patient burden. We have many patients that have been in multiple natural history studies over the years and still are awaiting a clinical trial, which might ask them yet to do another history study rather than leveraging what has already been established. Next slide. Some mechanisms that could be used to facilitate data sharing are patient advocacy-led consortia, pre-competitive collaborations, open data platforms like CPATH, scientific focus workshops for all stakeholders, and utilizing global advocacy networks such as TreatNMD. Next slide. We ask that you utilize regulatory flexibility tools to accelerate the development of therapies for the LGMDs. Placebo-controlled trials are unethical for patients who have irreparable progressive muscle wasting. Please provide guidance for sponsors on how to effectively utilize available natural history study data, real-world evidence, and biomarkers for surrogate endpoints. Time is muscle and is not on our side. Next slide. Sammy has waited 15 years for an approved treatment. She's grown to love children like Jackson, Chloe, Berkeley, Kennedy, and Jacob, who also live with LGMD. She's heartbroken for what their future will be like if left untreated. Please don't make these beautiful kids also wait 15 years and suffer the terrible losses like Sammy has. They deserve and need treatments now. On behalf of our entire LGMD patient community, thank you for the opportunity to share the urgent story of unmet need.
Great, thank you very much. Our next speaker is Nicholas Buss on behalf of BIO's Nonclinical Cell and Gene Therapy Task Force. Whenever you're ready.
Good morning, my name is Nick Buss, and I'm currently co-chair for BIO's Nonclinical Cell and Gene Therapy Task Force. I am also an employee of Eli Lilly. The Cell and Gene Therapy Nonclinical Task Force is a sub-team of BIO's Regenerative Medicine Committee. Next slide. Here are my disclosures. Next slide. The role of the Cell and Gene Therapy Non-Clinical Task Force is to identify key scientific and regulatory issues for cell and gene therapy products and work collaboratively to recommend appropriate science-based action. For example, we try to develop optimized non-clinical packages that reduce the number of unnecessary animal studies. Sometimes we work through modality-associated findings to accelerate our understanding, AAV-mediated DRG toxicities being an example. We also help to drive preclinical cell and gene therapy innovation and use industry knowledge for good non-clinical safety science and continue to provide an industry support network to collaborate with health authorities on topics as needed. Next slide. We use key learnings and knowledge to facilitate efficient development of cell and gene therapy products. For example, when animal pharmacology models are limited or unavailable, we encourage the use of in vitro models. We advocate for the appropriate reduction in the use of non-human primates for the development of cell and gene therapy products, ensure robust species selection for our studies, and appropriate study designs for the intended patient population. We also encourage modality-driven harmonized approaches where possible, for example, species selection, study endpoints, and study duration for AAVs. I would also like to emphasize that we request that non-clinical studies are conducted to the highest quality. Next slide. I would also like to say that interaction is critical for success in cell and gene therapy to support the preclinical challenges we often encounter. For example, efficacy and pharmacology studies support our reason to believe from a company investment perspective and are critical for researchers in early preclinical development. This can be very challenging even when there is a clear data-driven hypothesis. In the past, industry has received challenging requests for pharmacology studies that may prevent or delay development of cell and gene therapy products. We appreciate the support and opportunity for improved dialogue on challenges such as blocked access to models, inability to replicate published data, the lack of an animal model, or even the use of in vitro models. We also request that the agency continue to partner with industry on avoiding similar delays for future cell and gene therapy products. Next slide. Examples where we have been leveraging knowledge include justification for rodent-only GLP toxicity studies. With appropriate justification, it may be acceptable to conduct GLP toxicity studies in rodents. Rather than large GLP NHP toxicity studies, consider supplemental non-GLP short-term non-rodent studies only to characterize specific aspects and to aid clinical dose selection. We also like to acknowledge that this will be case by case and appropriate justification should always be provided. Other examples include the use of previously generated data, often referred to as platform data. For example, we can reduce the number of NHP studies by using previous biodistribution data, for example, with the same capsid serotype, or we can apply key learnings from non-clinical and clinical safety data, reduce and refine the number and extent of required toxicology studies. Such examples include the route of administration. For example, we have a better understanding of AAV-mediated ocular inflammation or AAV-mediated DRG changes in animal studies. Do we need to continuously demonstrate resolution for a known AAV-mediated finding by conducting more long-term NHP studies for each product? We can also refine the duration of toxicology studies. Studies up to 3 months are generally sufficient to characterize AAV-mediated findings. We can also reduce the number of interim necropsy time points, as often not informative. For example, an early 4-week time point could be omitted, where ocular imaging endpoints are as informative as ocular pathology. Next slide. And we encourage days like today. We need to maintain open dialogue with industry. We are very appreciative of opportunities to continue dialogue with the agency, as developing cell and gene therapy products for both rare and non-rare diseases is challenging and costly. We would like to work collaboratively to prevent requests for unnecessary non-clinical studies that can delay or prevent progression of products to the clinic. We encourage open discussion with industry and other health authorities, through example ICH, to align on non-clinical requirements, to utilize FDA expertise and databases to refine preclinical development. We'd like to continue to partner with industry and other health authorities in this mission to reduce, refine, and replace the number of animal studies used without impacting patient safety, and continued collaboration will reduce the cost of non-clinical development and ultimately benefit the patient. Thank you.
Thank you very much. Our next speaker is Daniel Grant on behalf of Novartis.
Thank you very much. My name is Daniel Grant. I'm from Novartis. I have no other financial disclosures. I'm currently leading a number of our AAV drug development programs, and I'd like to talk a little bit about some of the challenges we face developing gene therapies. Let's go to the next slide. So we believe that gene therapy has potential for broad application in the development of new medicines to treat a broad range of diseases. But the pathway to understanding the full potential of this new modality runs through rare monogenic disease right now. And that's where really almost all of our drug development in the gene therapy space is. And what that means is that most of our programs land at the nexus of this Venn diagram we're showing, which is that we are working in a space where we have rare diseases with a limited number of patients, very little precedent for what success looks like in the development of new medicines in this space. And on top of that is overlaid the complexity of working with a new modality, a new therapeutic modality. So what this means is that we find ourselves having to take novel approaches to setting up these drug development programs. And not only bringing new therapies to patients that have rare disease have been waiting for therapies for many years, but also our ability as a scientific community to access the full potential of this new modality is linked to our ability to move these programs forward despite the challenges that we're facing. Let's go to the next slide. So what does this look like from the drug developer's perspective? Well, we find ourselves working quite hard to strike this balance between practical considerations of setting up these programs, finding accessible patients, understanding how the community sees the disease, and then the scientific objectives that are associated with building an evidence package to support the development of a new medicine. Some of the kind of areas where we see these challenges really come to the surface are around the size of the studies that we're able to run. For example, oftentimes we find ourselves looking at the number of patients that we can include in a study and trying to understand what that means for our selection of endpoints, rather than the traditional way that we take this on, which is usually to think about our endpoints and then how to power the study. We find ourselves having to look at different types of data sources, non-traditional data sources, and then at the same time develop new endpoints and assessments to evaluate what success looks like in these diseases. So a couple of the things that we've learned on these programs that I want to kind of mention here. So firstly, engagement with FDA and with the patient community is far more important in the development of gene therapies and in rare disease than in other development programs. In addition, and really this is the reason that we put this presentation on the docket today, FDA engagements are incredibly important inflection points for these rare disease development programs where we're developing gene therapies. These programs often depend on our ability to find the right balance between practical considerations and scientific objectives and to get alignment with FDA on that balance. Thank you very much for the time.
Great, thank you very much. Our next speaker is Craig Malzahn on behalf of REGENXBIO.
Good morning, Craig Malzahn, EVP, Product Development and Chief Technology Officer at REGENXBIO. My disclosure is that I am an employee of REGENXBIO. REGENXBIO is a leading AAV gene therapy company. Thousands of patients have been treated with approved and investigational gene therapies built on our NAV technology platform. We are advancing a late-stage clinical pipeline of gene therapies for rare and retinal diseases. Next slide. I'll focus on our RGX-202 program for Duchenne muscular dystrophy to highlight how we've integrated learnings from other gene therapy programs and a CMC platform approach, and how these can contribute to a favorable safety and efficacy profile for patients. RGX-202 is being evaluated in the pivotal Affinity Duchenne trial and was designed with real-world evidence-based patient needs and commercialization in mind from the outset. We implemented end-to-end fully integrated processes, enabling seamless transition from research lab to clinical development to commercial manufacturing. We believe this integrated approach contributes not only to the profile of RGX-202, but also streamlines the BLA review process. Next slide. Gene therapies for Duchenne use a microdystrophin or smaller version of the dystrophin protein, but not all constructs are created equally. The 202 microdystrophin construct includes the CT domain, a key part of functional dystrophin, making it the closest to naturally occurring dystrophin. Published preclinical data demonstrated clear functional benefits from including the CT domain, such as higher levels of microdystrophin protein expression, increased muscle force, and improved resistance to damage. Next slide. There are well-documented potential safety events associated with current and prior high-dose AAV gene therapy. And that is exactly why we took learnings from these programs and implemented a proactive immune suppression regimen designed to specifically counter known liver and complement issues from other programs for consistent improved safety outcomes. And from the data we've reported from the phase 1/2 trial, we know this approach is paying off. This proactive short course regimen includes a combination of corticosteroids, sirolimus, tapered eculizumab, which is a C5 inhibitor, and prednisone based on weight, all concluded by 3 months post-dosing. Today, boys with Duchenne only have one opportunity for gene therapy. That's why we took the safety data from the field and designed this program to maximize the opportunity for functional benefit while importantly reducing the risk of adverse events. Next slide. This data-based immune suppression regimen, combined with our industry-leading manufacturing, may contribute to the favorable safety profile seen in patients receiving RGX-202. 202 uses our best-in-class NavXpress suspension manufacturing platform. This is a consistent, high-yielding, automated, and scalable process that is delivering a very high full capsid product purity profile, which translates to a higher efficacious dose, enabling a lower total viral load. Using a manufacturing platform offers significant advantage in applying learnings from prior programs to further enhance the 202 product profile, as well as providing a uniform framework for the CMC sections, where prior agency feedback is incorporated upfront, enabling a more streamlined BLA approach. Next slide. Now, I'll highlight how these elements of the novel construct, proactive immune suppression, and manufacturing are translating to the clinic. As reported in the phase 1/2, RGX-202 was well tolerated with no SAEs or AESIs, including no signs of liver injury. Additionally, we see robust transduction and expression of the 202 microdystrophin. Patients demonstrated functional improvements, which were meaningful compared to natural history external controls at 12 months. Next slide. In a heterogeneous and progressively debilitating disease like Duchenne, using baseline matched external natural history controls provide a clear picture of functional benefit. On the left, you can see the established disease trajectory of Duchenne. Patients slowly gain skills and plateau before reaching a period of rapid decline. On the right, you can see the positive improvements on time function tests from 4 participants 1 year after receiving 202, compared to how they'd be expected to perform without treatment, based on a matched natural history control group. Not only is there an established strong scientific basis in using matched natural history controls to evaluate drug effect, but this method also enables accelerated drug development for patients facing great unmet need. To close, thank you for the opportunity to share an example of how we've effectively integrated data and learnings from multiple disciplines into our programs for the ultimate shared mission of serving patients. Thank you.
Great, thank you very much. Our next speaker is Bruce Levine on behalf of the International Society for Cell and Gene Therapy.
Hello, everyone. Thank you for the invitation and the opportunity to present. I'm a past president of the IACT, current chair of the Committee on Ethics of Cell and Gene Therapy, and hold a faculty position at the University of Pennsylvania. Next slide. The IACT has been established in 1992. We are a global society of over 4,500 clinicians, regulators, researchers, technologists, and industry partners. We share a vision to translate cell and gene therapy into safe and effective therapies to improve patients' lives worldwide. Next slide. I have a financial interest due to intellectual property and patents in the field of cell and gene therapy. Those are listed here. Conflict of interest is managed in accordance with the University of Pennsylvania policy and oversight. Next slide. In March 2018, at a symposium on cell and gene therapy manufacturing at Stanford University, I presented this slide detailing the absence of genotoxicity in T cells with retroviral and lentiviral vectors. Those publications are listed, and I advocated for a revisiting of the test requirements for retroviral and lentiviral transduced T cells. Next slide. Next. This is the experience at the University of Pennsylvania and Children's Hospital of Philadelphia. This was a safety analysis in 783 patients treated with retroviral and lentiviral modified products. Next slide. This was a compilation of 38 gene-engineered T-cell therapy trials targeting HIV or cancer, more than 2,200 patient years of observation. There was reports of secondary malignancies in 18 patients with a median onset of 1.94 years with solid tumors most frequent. Importantly, there was no evidence of insertional mutagenesis or high-level CAR marking observed, and no T-cell malignancies that could be attributed to the cell therapy. Next slide. More details from this report that appeared in January. Vector integration site analysis was done on a subset, 169 patients. No pathological insertions linked to secondary malignancies. In some cases, there was integration in or near specific genes associated with monoclonal expansion and sustained T-cell persistence, and I'll come back to that later. Next slide. Our experience in producing vector lots, more than 33 at this point, no replication competent lentivirus observed, more than 1,000 clinical trial gene-modified T-cell products, no RCL, 25 years of gene-modified T-cell experience, no RCR or RCL. Other centers report the same. The NIH has the most experience with more than 35 years of gene-modified T-cell experience and no RCR. And I'll just point out that we've been requested to provide stability data for vectors using the same backbone, but merely simply swapping out the CAR transgene. And I'll have to ask, is the time and cost justified given the previous decades of safety data? Next slide. In 2012, we reported decade-long safety and function of CAR T cells in HIV. This was a compilation of 3 clinical trials using a first-generation CAR. Next slide. So there have been reports of clonal expansion, and the question is this an oh no event, or is it a that's strange, what's happening? Next slide. And advance next. And one more. Thank you. So this was a clonal event first detected by flow cytometry of TCR-V beta families. It turned out that this patient had an integration in a gene called TET2 and a naturally occurring mutation in the other allele. TET2 is associated with stem cell renewal in hematopoietic stem cells. What I want to point out here that you can see in red on the right is there is this emergence of clone, but with disappearance of the target, meaning CD19 positive B cells, that resolves, and there's the emergence of an oligoclonal population. Next slide. This is a second report from Stanford with an integration into the Sybil gene, also triggering proliferation in a CAR22 product. And again, the same thing happens. There's a oligoclonal, almost clonal population, but then that resolves. Next slide. And so we know that
excuse me, I'm sorry, Dr. Levine. I'm going to have to interrupt you. Unfortunately, we have to move on to our next speaker, but we encourage you to please add the remainder of your presentation to the docket. Thank you very much.
Okay, thank you.
Okay, great. Thank you so much for those comments. Our next speaker is Sumarendra Mohanty on behalf of Nanoscope Therapeutics.
Good morning, and thanks to CBOR for the opportunity to speak. I'm Samar Mohanty, co-founder, president of Nanoscope. I'm an inventor and have financial interest in the company. Today, I'm excited to share our groundbreaking work in bringing the light of hope for patients with severe vision loss. Next slide, please. We are leveraging 5 critical knowledge areas to facilitate development of fast disease-agnostic in vivo gene therapy to restore vision. These represent a paradigm shift from the traditional gene-specific approach. By recognizing the shared pathology across different diseases, we are paving the way for mutation-agnostic gene therapy. At the same time, we have been leveraging heterogeneous real-world data to establish clinical meaningfulness of our endpoints. We are also exploring how safety and efficacy may be influenced by pre-existing conditions across indications. Throughout the development, transgene expression data have been fully integrated to inform our decisions. Ultimately, our goal is to build a disease-agnostic platform for neurodegenerative eye condition, allowing us to apply safety and efficacy insights across multiple indications. Next slide. Let me paint the picture of why this approach is so desperately needed. The challenge is that the IRDs represent the most complex therapeutic areas in medicine today. We are dealing with over 8,000 mutations in more than 250 genes that have been associated with IRDs. And remarkably, 1/3 of these mutations remain unknown. Current gene therapies are limited to patients with precise genetic diagnosis. This leaves the vast majority of patients with no treatment options, especially those with very rare genotypes. These approaches also become ineffective when the target cell, the photoreceptors, is not present in the advanced stage of the disease. Next slide. Next slide, please. Here is where the disease-agnostic approach like ours can solve these challenges. Despite the incredible genetic diversity underlying IRDs, there is commonality in the disease progression pathway. Whether we are dealing with RP, Stargardt, choroideremia, the end result is same: progressive degeneration of photoreceptor leading to vision loss. This shared final pathway of cell death represents our therapeutic opportunity by targeting the bipolar cells that are preserved in these conditions and delivering to them the tools to restore vision. Next slide, please. To do this, MCO10 therapy combines the best from gene and cell therapy approach. Instead of trying to fix the broken genes or replace the dead photoreceptor, we are reprogramming the surviving retinal infrastructure. MCO10 works by delivering proprietary engineered opsin to non-light sensing surviving bipolar neurons, transforming these cells into photoreceptor. A single intravitreal injection reprograms the retinal circuitry, allowing patients to regain vision regardless of which gene or disease caused the blindness. Next slide. The second critical area where we are breaking new ground is establishing clinical meaningfulness through real-world data analysis for over 70,000 RP patients in the AO registry, which shows that natural progression results in a decline of 1.5 letters per year. For the first time in a randomized trial in RP patients, we showed 0.3 logMAR improvement equivalent to 15 letters. That means a vision that is lost over 10 years is gained on average. This durable improvement has been observed over 3 years and expected to continue. Next slide. We are also trailblazing in understanding how pre-existing conditions affect treatment outcome. Most patients have neutralizing antibodies against AAV from childhood exposure. Yet, remarkably, our studies indicate that MCO10 therapy improves visual acuity regardless of baseline NAV values. We have treated patients across dozens of mutations and with therapeutic doses with no reported SAE. Importantly, the patients had evidence of improvement irrespective of gene mutation. And looking holistically across endpoints, those who did not improve in visual acuity had evidence of improvement in mobility or self-discrimination. Next slide. The fourth area where we are making advancement is in integrating transgene expression data throughout the product development. We are using red fluorescence imaging to track gene delivery to target cells and measure the retention across cells, animals, and humans. This comprehensive approach allows us to objectively determine the potency and optimize dosing across development cycle. Next slide. Finally, our disease-agnostic platform allows us to leverage safety and efficacy data across multiple neurodegenerative conditions of the eye. Our RP clinical results show statistically significant visual acuity improvement as compared to controls. When looking across endpoint, most patients have evidence of improvement in 2 out of 3 key efficacy assessments, demonstrating the broad functional impact. Importantly, all subjects had evidence of improvement in either of the three key essays, and the majority of MCO subjects improved in visual acuity. To summarize, our disease-agnostic approach represents a paradigm shift in treating retinal degenerative disease with a universal platform, and the implications are far beyond retinal degenerative disease. Thank you for your attention.
Great, thank you so much. Our next speaker is Nancy Myers on behalf of Catalyst Healthcare Consulting.
Good morning. My name is Nancy Bradish Myers, and I'm CEO and founder of Catalyst Healthcare Consulting, a regulatory strategy and policy firm that's worked for over 20 years with global innovators, academics, patient groups, investors, and trade associations to advance regulatory policy, filing strategies, and patient access. My financial disclosure is that as a consultant, I get paid by a number of groups to think about these types of policy issues. My remarks today come from the vantage point of a consultant who sees across multiple pipelines and perspectives. Next slide. As this slide shows, knowledge can be leveraged at several levels, each with different implications. It can be sponsored leverage by sponsors leveraging their own data, knowledge sharing across partners like CDMOs and CMOs, and broader cross-stakeholder knowledge aggregation. As CBER continues to consider how to best support the use of prior knowledge, it will be important to seek stakeholder input on actions before they are implemented to ensure policies do not undermine innovation. Next slide. There's a broad agreement that sponsors should be able to reference their own data across cell and gene therapy assets with similar characteristics. CBER can support this action by 1, following cross-referencing to a company's own BLA, not just NDA, as currently limited by the Platform Technology Designation guidance. This approach would save both sponsors and reviewers resources. 2, establish a process that allows a sponsor to leverage existing knowledge of data across similar products where appropriate. For example, full biodistribution studies may not be necessary for products using the same predictable delivery platforms like LNPs. Note, knowledge sharing can also cut down on the use of animal models if a single study can support multiple programs. 3, CBER could take simple steps that signal how to advance things by adding an FDA-led discussion point in early meetings like Interact and pre-IND meetings to explore how sponsors might leverage prior knowledge. It's a subtle action, but it will signal FDA's interest in efficiency. Next slide. The next level of leveraging involves sharing knowledge across external partners. FDA should consider revisiting the 2024 final rule that prohibits BLAs from cross-referencing drug master files for CMOs. Permitting this cross-referencing would reduce duplication of certain information while protecting CMO proprietary methods. Next slide. The FDA's pivotal role in aggregating and sharing regulatory learnings is critical, but any steps need to first do no harm and avoid undermining first-in-class innovators. 1, I encourage OTP to continue publishing its evolving expectations around common challenges through formal guidance and other written means. Also, town hall discussions in 2023 and 2024 were seen as a success. I am glad to see another one scheduled on CMC and urge these interactions become more routine. 2, please participate. It's important for CBER staff to attend and speak at conferences, workshops, and scientific exchanges, not just remotely, but in person. Direct engagement builds trust and allows for productive dialogue, which will in turn lead to improved reviews. Industry and stakeholders appreciate an agency that fields questions, encourages dialogue, and seeks alignment. 3, reevaluate and adapt. I suggest FDA create a streamlined process that encourages stakeholders to appear before FDA, specifically CBER for topics with cell and gene therapy, to jointly present new data and suggest updates to outdated requirements. For example, building on a little bit what you heard from Dr. Levine, my team and I have helped lead a multi-stakeholder effort to draft an article currently in preprint form that advocates for the reevaluation and shortening of CAR-T therapy's long-term follow-up requirements and calls for modernizing data collection. The collaboration is a model for how the field can evolve when science, real-world evidence, and alignment around solutions come together. In closing, as cell and gene therapy matures, next slide, we must preserve predictability, fairness, and an ability to get patients treatments. We need to ensure confidential and proprietary information remains protected, especially as new tools like AI are deployed in reviews. And we need to ensure efforts done in the name of transparency do not bypass the checks and balances built into the system. And I'm referring to the CRL issue. Thank you for this opportunity to speak.
Great, thank you very much. Our next speaker is David Schmall on behalf of Foundation for Accreditation of Cellular Therapy.
Thank you. I'm David Schmall. I am the CEO of FACT, and I have no financial disclosures to make today. Next slide, please. My objectives today are to describe who FACT is, what we offer, and the value of those, to highlight some recent engagement of FACT with the FDA through the American Society of Transplant Cellular Therapy's 80/20 Task Force, and to showcase action taken FACT is making to bring these curative therapies closer to eligible patients. Next slide. FACT offers three core things to our community and stakeholders. First is a peer-developed standards developed by a consensus-driven process. These establish minimal guidelines versus best practice of quality and safety and are dynamic and frequently updated. Based on those standards, we offer a voluntary accreditation process that is modular in nature, which I'll describe in a moment. And then supporting all these education tools and services for provider programs to understand and prepare should they choose for our accreditation process. Next slide. We were founded over 3 decades ago by providers and for providers. The leadership of what is now the American Society of Transplant Cellular Therapy and the International Society of Cell and Gene Therapy, envisioning that a built-in mechanism for quality and safety would protect patients through the entire transplant and cellular therapy journey. We anticipated the future expansion of these therapies that we're now seeing today, striving for efficiencies, and of course, complementing existing regulations and laws. Next slide. This slide showcases the 4 bodies of published standards we have today, dating back to 1997. Our HCT standards are now in their 9th vision, and our IEC, for example, are in their 3rd vision to be published this fall. Next slide. The publication and development of our IEC standards back almost 10 years ago demonstrates how FACT has evolved as these cellular therapies evolve through time. And this is an article in the Journal of Immunotherapy of Cancer citing that a robust quality infrastructure is key to safe and effective delivery of IEC cells and how FACT can help. Next slide. Moving from our standards and our accreditation, this slide demonstrates the various services that programs can apply to receive FACT accreditation, either in total or in a modular nature. The accreditation is designed to be flexible depending on the services rendered in a provider setting. Next slide. And the value of our voluntary accreditation offerings, commonly respected and trusted as a level of quality and safety. Patient assurance, an assurance of quality and safety among all other stakeholders in the field. This is administered by FACT, which is nonprofit and unbiased, and through a peer-based community of experts and trained inspectors, and ultimately driving standardization and corresponding efficiencies for all involved. Next slide. By the numbers, you see the number of accredited programs through FACT. totaling 200 currently, and then broken down to IUC and the allotransplant programs. Next slide. Recently, the American Society of Transplant Cellular Therapy formed an 80-20 task force, which has consistently upheld and demonstrated the value of FACT. It was first convened in 2021 to standardize and streamline requirements for treatment center onboarding and the maintenance of operations for these cell therapies, particularly CAR-T. Next slide. After that first convened meeting, two outcomes were produced. First was the consensus agreement to advocate FDA for the continual reduction and ultimate elimination of REMS. And then the 5 consensus-based statements you see on the right, and I'm highlighting that existing professional accreditation groups such as FACT should be used for most oversight and not the manufacturers. Next slide, please. Following that first convened workshop, 2 other events happened that were material for the 80-20 task force. First, in 2022, the FDA convened members of the 80-20 and also FACT, and this was focusing once again on sunsetting REMS moving forward. And then in 2023, the second large workshop occurred with 70 attendees, publishing a statement that says consensus of professional society guidelines, central accrediting, and auditing bodies such as FACT and NMDP could be leveraged to ensure safety and quality practices in the absence of REMS. Next slide, please. That led to this published paper titled Awakening from REMS, ASTCT 80-20. Next slide. And these quotes, once again, arguing for the support of FACT as an independent society-based accrediting body. Finally, moving forward. Next slide. We're taking action at FACT to bring CAR-T therapies closer to where patients live. We will be publishing fit-for-purpose standards for community sites, investing in headquarter resources to support the accreditation, and offering FACT consulting services to help these community programs navigate the accreditation. And finally, participating in multi-stakeholder organizations such as the CAR-T Vision Steering Committee and Act for Hope. Thank you very much.
Great, thank you so much. Our next speaker is Lana Shu on behalf of Kite Pharma.
Good morning. My name is Lana Shu, and having spent over 10 years as an FDA medical officer earlier in my career, I appreciate this opportunity to share my perspective today as the global head of regulatory affairs at Kite Pharma, and I have no other financial interest. Next slide. Today, I will talk about how the field can leverage growing knowledge as more real-world evidence become available to use this information to inform regulatory adaptations. I will focus on the long-term follow-up for CAR T-cell therapies. The original 15-year requirement was based on the theoretical risk of insertional mutagenesis set at a time when very little real-world evidence and data existed. Next slide. So now today, we have nearly 15 years of experience and over 30,000 patients treated. This collective experience shows that CAR-T has a well-established safety profile. Next slide. To evaluate this evidence, we have brought together 21 authors for this white paper, including voices from the patients, the academia, the industry, and the NIH, the National Cancer Institute. Next slide. And briefly, our process included direct inputs from the patients and the providers, literature review, and new anonymized composite clinical trial safety data. Next slide. And here's what the mandatory follow-up safety data do show. On the left, most adverse events are reported within the first year post-infusion of CAR-T. Adverse events reporting sharply decline after year 1. After year 5, reports of events are rare and approaching 0. Thus, keeping a study open for another 10 years will not yield meaningful safety signal when no reports are coming in. On the right, 13 published studies from 2018 to 2025 showed that second primary malignancies after CAR-T treatment are similar to the background rates as if you received standard of care treatment. T-cell malignancies are rare, and causal link to CAR-T has not been established. Next slide. So when key adverse events do occur, they occur early. So the FDA's own data showed that T-cell malignancies occurred within the first 2 years. Published studies showed that most malignancies occur within the 5 years. And University of Pennsylvania have been using CAR-Ts for over 25 years to treat oncology, HIV, pediatrics, autoimmune patients. They had data from 783 patients and found only one case occurring after year 5. It was a thyroid cancer at year 14, occurring in a patient with previous history of radiation to that area prior to receiving cell therapy, CAR-T therapy. Next slide. So we also looked at how data collection could be improved. Today's labor-intensive manual method of data collection can be modernized by automating AE capture directly from the electronic health record. CBER has conducted an initiative called BEST, which has already taken some promising steps. They piloted an automated AE collection for vaccines, which resulted in regulatory-grade data quality, and also explored using artificial intelligence for signal detection. Next slide. So based on these findings, we have our recommendations. These are the following. 1, we could shorten the long-term follow-up to 5 years for CAR-T therapies, those that use integrating vectors. Adaptively, agency can truncate or extend based on what is seen in the first 3 to 5 years of follow-up. Secondly, we can modernize AE data collection using electronic health record, which could enable comprehensive safety data collection even beyond the long-term follow-up period, and thus decreasing the burden on the healthcare providers and on the patients. Thirdly, we could focus the data collection on events that are clinically meaningful and mechanistically relevant. Next slide. In summary, the data supports streamlining long-term follow-up requirements for CAR-T cell therapy. Next, we have, initiative to modernize using electronic and artificial intelligence tools to enhance safety monitoring. This is the model for adaptive regulation. We could balance agility with accountability and aligning with patients' needs. Thank you.
Great, thank you very much. Our next speaker is Pushpendra Singh on behalf of Ocugen.
Good morning, everyone. I'd like to thank the organizer for the opportunity. My name is Pushpendra Singh. I am the head of product development here at Ocugen. And at Ocugen, we are bringing the transformative therapies for the patients in need, particularly for the gene therapies, which are gene agnostic, and for the ocular disease indications. So as we know, the cost, next slide, please. As we know, the cost of the therapies are quite high. They are millions of dollars. So the question remains that how we can actually make them affordable, and how can we actually bring it to the patients in need much earlier, which is the need here. So as you see the breakdown here in this Venn diagram, so the cost for any therapy is actually derived from the manufacturing, clinical trial, and the upfront R&D work that has been done there. So as seen in here, so 40% is contributed from the manufacturing, about 18% from the clinical trial, and 23% from the discovery and preclinical work. Of course, this can vary from one indication to one therapy to another one, but this remains the same, more or less. Next slide, please. If we look at it more closely, the gene therapies are costly because of the complex manufacturing, because AAVs, lentiviruses, they are made under the strict GMP requirement, and number of campaigns are done in order to bring the therapy to the market. There is a high upfront R&D cost, decades of work. The preclinical, both as in the process actually goes, which is having a lot of failure rates, and that's where the cost actually is spread over to the patients. There are stringent regulations which are to actually ensure safety, potency, and the stability, and that is mandated by the regulators. Often, in case of gene therapy, you can produce only so much, like the small batches produced, and in some cases, you might have to have multiple batches to produce so that you can actually provide it for few patients. And one important thing remains that clinical trials for the gene therapies are kind of challenging, so that actually affect the global recruitment. Often they are indicated for the rare disease, and it's become hard to actually find the patients. And in addition to that, there is a long-term follow-up burden, and that really cause the cost to high, this 10 to 15 years of follow-up. Next slide, please. So, I mean, the way I'm thinking about it, I'm trying to actually put the thoughts in here to reduce the cost and improve affordability for the gene therapies. Particularly, there could be some there could be some innovative approaches. There could be some modular GMP sites which can be created that can provide the forum to produce the high vector yield in a scalable bioreactor manner. For AAVs and the lentivirus, these are the platform, and the approved, so whatever approved BLAs are there, that knowledge can be shared and that can be applied for the developing therapies, and that will reduce the requirement of the studies, and hence the cost can be brought down. Automation optimization is always the a need for any therapy, and there could be global hub generation creation which can decentralize the sites near patients, and that can reduce the logistics of bringing the gene therapy that often require the ultra cold temperature storage. And we can take some learning and can apply some learning for the clinical trial from the innovative models which help the recruitment faster for the patients. So they are like some examples, like China's ITTs are there, Australia's CTNs are there, and in addition to that, we can have some shared facilities in the vector supply, especially that will do a lot of good for the rare and ultra rare disease. And lastly, there could be the pricing decided based on the outcome in the patient, and that can be broken down into the number of installments and subscription model for the affordability. With this context, next slide, please. With this context, actually, so there are several questions that I think I echo with a lot of speakers here, and are trying to see that what FDA can play a role in reducing the cost and accelerating these therapies. So number of things can be thought through, like there's a requirement of the modular GMP suites, which can hugely cut down the manufacturing cost. There could be less burdensome regulatory pathway where you can non-clinical and the process data can be shared across the different related indications, and leveraging the non-clinical and the process data can, or minimal bridging data can really help to justify some of the studies that you can do the risk assessment on those part. And will FDA be okay to actually have the hospital-based dosing after IND clearance, similar to Australia and China model? And lastly, the regulatory fee can be kind of waived off. And so these are some considerations. Again, this is not an exhaustive list, but this is something I like to actually have thought on. Thank you.
Great, thank you very much. Our next speaker is Chris Williams on behalf of the National Institute for Innovation in Manufacturing Biopharmaceuticals.
Hey, good morning. I'm Chris Williams, an independent consultant registered as CR Williams Consulting LLC, speaking on behalf of the National Institute for Innovation in Manufacturing Biopharmaceuticals. NIMBLE is a manufacturing USA institute operating out of the University of Delaware and funded through a cooperative agreement with the National Institute of Standards and Technology and the U.S. Department of Commerce, with significant additional support from its member organizations. NIMBLE is a nonprofit public-private partnership founded for the advancing of biopharmaceutical manufacturing, solving industry challenges, and developing the skilled workforce to enhance the competitiveness of the United States in this sector. Since its inception, NIMBLE has funded and facilitated dozens of pre-competitive collaborations to overcome the biggest and most prevalent challenges in biomanufacturing and related technologies. For the purposes of this public listening session on leveraging knowledge for facilitating development and review of cell and gene therapies, I want to contribute NIMBL's efforts in the area of viral vector gene therapies. Next slide, please. NIMBL leads a viral vector manufacturing and analytics program. The program enlists more than 40 volunteer individuals from member organizations who are subject matter experts across the relevant scientific disciplines. Each individual's time is sponsored by their respective NIMBL member organization. Representation ranges from pharma, CDMOs, leading academic institutions, technology suppliers, and relevant federal agencies. NIMBL uniquely brings together these experts to collaborate pre-competitively on shared challenges in the manufacturing and analytics of adeno-associated viral vectors, commonly used for clinical and commercially approved gene therapies. NIMBL as a body, acting within our mandate, and our volunteer individuals as representatives of the scientific community recognize the economic challenges in developing, manufacturing novel gene therapies at a cost that's sustainable to society. In response, the NIMBL Viral Vector Program has committed to leveraging our combined expertise to furnish an open access AAV CMC platform to be shared, referenced, leveraged, and applied by any sponsor. Next slide, please. NIMBL is currently investigating into an assemblage of critical materials, standard process operations, and analytical tools to move a novel AAV gene therapy into phase 1 clinical trials. Following internal development and demonstration, these will be made publicly accessible for the purposes of drug development by any independent and unaffiliated sponsor. Leveraging platforms, especially decentralized open access platforms, will become beneficial to sponsors, manufacturers, and regulators. Diluting costs, avoiding rework, reducing risks, and saving time all contribute to a more positive outlook for the CMC development of novel gene therapies. We believe that facilitating this approach will make developing unique gene therapies for smaller patient populations more feasible, invigorating investment in the sector, and thus making the potential therapeutic benefits more broadly accessible. Next slide, please. The NIMBL Viral Vector Program and I personally see these opportunities for sharing information for the mutual benefit among sponsors in 2 stages. First, when it comes to developing novel gene therapies, NIMBL and its members are working to enable sponsors to share knowledge on common elements in vector design, manufacturing, and testing. Within NIMBL, we are furnishing a vector CMC platform that will be openly available for independent sponsors to use in this way. We believe that such a standard platform also provides a basis for shared understanding across the ecosystem, from sponsors to technology providers to health authorities. And second, we'd like to encourage sponsors to consider how they would share their real-world evidence with the motivation of improving CMC processes for future therapeutics. Drawing connections between therapeutic design, manufacturing, quality, and ultimately patient outcome is a challenge worth pursuing. This may only be powered by sharing real-world evidence of many therapeutics that have been developed on a common framework. And in that vision, NIMBL believes an open access vector CMC platform can help enhance the ecosystem for developing novel genetic medicines. NIMBL, as a nonprofit public-private partnership, can provide a neutral ground, legal frameworks, and standards to make data sharing safe and valuable for all parties. Thank you.
Great, thank you very much. Our next speaker is Valerie Winslow on behalf of Sanofi.
Good morning, everyone. I'm Valeria Winslow, Global Regulatory Lead at Sanofi and Head of Regulatory Gene Therapy Team. I'm here today to share Sanofi's vision and progress on gene therapy development and briefly address some of the FDA questions. Today, my remarks are my company position. The Genomic Medicine Unit at Sanofi is a program-driven platform that is at the forefront of the gene therapy innovation. We have brought together experts from different fields, including platform development, translational science, and manufacturing, to develop new gene therapies across multiple therapeutic areas. Historically, Sanofi has focused on rare single gene disorders. However, we are now strategically expanding our approach to address both rare diseases and more common disease areas, aligning well with the FDA interest in broadening therapeutic applications for gene therapies. Sanofi currently has several active INDs spanning multiple gene therapies technologies and disease areas. These give us a substantial experience in navigating regulatory pathways and generate valuable cross-program insights that inform our current regulatory science. We are establishing cutting-edge platform technologies approach, both for viral and non-viral gene therapies modalities. At Sanofi, we view platform technology as fundamental to the future of cell and gene therapy. Our current experience shows that well-characterized platforms can significantly accelerate IND-enabling studies, demonstrating the potential to simplify the development and the regulatory process. In this regard, we welcome further FDA guidance on the eligibility criteria and the standards for demonstrating significant efficiencies in platform approaches. We propose further collaborative discussion on the practical implementation of platform designations across the product lifecycle. On the company position regarding the importance of data sharing and external collaboration, Sanofi recognized the significant advantages of sharing pre-competitive data and fostering collaboration across industry, academia, clinicians, and federal partners. This strategy perfectly aligns with our commitment to data transparency while ensuring the protection of our proprietary information. We believe that this approach could streamline research processes, accelerate development, improve data analysis, and ultimately enhance treatment safety for patients. Sanofi takes a proactive approach in sharing the results of our preclinical and clinical investigations with the scientific community, regulatory agencies, and patient advocacy groups. Currently, we are collaborating with a real-world evidence consortium to harness real-world data and with the DMCRN platform. The goal of these strategic partnerships is to generate regulatory-grade real-world evidence for hematological and neuromuscular conditions, allowing us the development of robust study designs and facilitate the identification of clinical meaningful endpoints, ultimately supporting regulatory decision-making and improving patient outcomes. We also see great potential for collaboration in the implementation of the long-term follow-up studies. Collaborations using AI-driven predictive modeling capabilities in this area have the potential to revolutionize long-term safety monitoring and enable the detection of solid safety signals across the diverse patient populations such earlier than traditional methods. In closing, on behalf of Sanofi, I would like to express our gratitude to the FDA for the opportunity to give insights as you consider future directions and regulations around cell and gene therapies. We look forward to continued collaboration and innovation in this exciting field. Thank you.
Great, thank you very much. Well, that was a fantastic session. I'd like to thank all of our speakers for all of those important insights, very helpful to us. We will now go to our FDA panelists to see if they have questions for any of our session one speakers. So I believe Dr. Young-Wook Choi has the first question.
Thank you. My name is Young-Wook Choi, a bioinformatics reviewer in OTP. I have a clarifying question for Nancy Myers. So one of the bullet points in your slides mentioned reducing the number of off-target assays. So for example, in CRISPR-Cas9 system, are you referring to leveraging off-target data across different guide RNAs that target different genomic loci, or leveraging data for the same guide RNA across different products?
I think it could be done several different ways. I think where I was focused the most was on LNPs, and kind of thinking that if you've had experience already and you've started, you your data shows that, there are many off targets that are unnecessary, then it would then be where you could try to start focusing the types of information that you have to collect. I think that throwing open the doors and looking at everything all the time probably is not the most efficient approach. So I think that's where I was headed.
Okay, thank you.
So thanks.
Great, thank you very much for the question. So our next question is going to come from Dr. Emily Wires.
Hi, my name is Emily Wires. I am a farm tax reviewer. We actually have two clarifying questions for Nicholas Buss. The first is, could you please clarify if your proposal is that the reduction and elimination of the interim necropsy time points would be based on pilot non-clinical studies with the intended clinical product, or leverage from products in the same class administered via the same route of administration?
Yeah, no, thank you. It's a good question. I think it's a combination of both. So, for example, if you have pilot tox studies where you have duration is say 4 weeks, then that data can support the GLP tox study if it's been sufficiently run to appropriate standards. I think if you do not, for example, have the pilot tox or the dose range finding, I think there may be occasions where you can omit an interim necropsy based on the endpoints you include in your study to monitor for known AAV-mediated toxicities, for example. And the example I gave is, for example, if you are injecting into the eye, you have a whole plethora of imaging endpoints that you can use, OCT, ERG, ophthalmoscopy, which is probably a more robust longitudinal assessment of ocular toxicity than having the need perhaps to do a necropsy at 4 weeks.
Okay, great, thank you. And then another question. So regarding the justification, what would be needed to justify the use of rodents to inform the dosing and safety of an AAV product?
Again, this is, as mentioned, it's very much case by case, and I think you would have to use platform data you have. For example, an experience with capsids. I think you have to look at the animal model, your indication, and I think the route of administration. And if, for example, and the fact that a rodent is a relevant species, then I feel like the rodent should suffice. Again, it's very case by case. I think if you're looking for it may be important to include a small non-human primate study, for example, to offer supporting data to help bridge between rat, monkey, and human. But then if that data is available, say on another project with a capsid, similar promoter, for example, then I would imagine that could be used as well.
Okay, great, thank you. We actually have one more question for Pushpenda, Singh from Ocugen. So could Ocugen give an example of what justification would be needed to minimize bridging non-clinical animal studies, and what kind of bridging data would be needed?
Yeah, I think, thank you for the question. So I think for the non-clinical data and the bridging of the non-clinical data, let's say if we are going with the similar indications, let's say the indications are in the ocular and there are many, developing therapies are there. So one thing is that we are producing, let's say, multiple batch of the different gene therapies for 2 different indications. So in that case, if we are using the same manufacturing, I believe those whatever non-clinical studies have been conducted with the same dose of administration, so that they can be simply taken for the biodistribution as well as for the toxicity. So because the capsid serotype remains same, so and the manufacturing process remains same, so that can be directly utilized and leveraged to go forward for the IND submission. With that, you can do the risk assessment. So that's one. But I'm saying across globally also, let's say there are different sponsors are producing the data. So is there a mechanism that we can come up with that all those redundancies can be cut down? So you have somebody has done worst case scenario highest dose, and if you are going with the lower than that, can that be utilized even from the other sponsor? So that will be something path breaking, and that can really cut down the cost on that aspect.
Okay, great. Thank you.
Thank you.
Great. Thanks so much. Okay, let's see if we have any more questions from our FDA panelists. It looks like we do not.
This is Katarina.
Oh. I hear someone speaking. Oh, Katarina. Great. Go ahead.
I have to our meeting for the greater value for. I have a couple of one question for Dr. Mohanty from Nanoscope. I understand that you find it useful to share safety and efficacy data among different indications for your product. But since this product, since this optogenetic treatment is a category of its own, would you find interesting to leverage data among different products of the same category too?
Thank you for the question, but sorry, I could not hear it very well because of some audio issues. Can you please repeat the question, Dr.
Yes, of course. I am going to use my headphones. Okay. Can you hear me better now?
Yes, please.
Okay. So I understand you would like to leverage data among the safety and efficacy data for your product on different indications. But since your product is a separate category on its own of genetic treatments, would you find it useful to also share data among products of the same category? So leverage data among products of genetic treatment products as well.
Yes, not all optogenetic products are same. Our opsin is very broadband and highly sensitive and targeted to specific promoter, bipolar cells. So across indications in ocular space, I think the data can be leveraged. But in future, if this probe same opsin is used for neurological indications, for example, pain and other neurological conditions, then it should be leveraged that the expression of the opsin is not creating any immune response or things like that. So, but broad from an optogenetic category, I think all opsins are different. Our opsin come from 3 different non-mammalian proteins, is a fused protein. So I'm not sure if we can share data across different developers. But of course, I will say that neutralizing antibody baseline value, how it impacts the efficacy or safety to a AAV2 serotype, our data can be leveraged to other manufacturers.
Thank you very much.
Thank you.
Great. Thanks so much. I'm wondering, do we have any more questions from our FDA panelists? I'll give a couple of seconds to see if anyone has anything they want to ask. Okay, great. It looks like we do not have any more questions. So at this point, I'd like to thank our FDA panelists and all our public speakers in session 1 for your preparation, your presentations and remarks. We're going to break for lunch, and we will resume the public meeting at 12:45 p.m. Eastern Time. If you are one of the public speakers in session 2, we ask that you please return to the meeting at 12:30 p.m. Eastern Time. At that time, you will be given access to the microphone and camera. Thank you all again, and we'll be back at 12:45 p.m. Thank you.
Welcome back to our public listening meeting on leveraging knowledge for facilitating the development and review of cell and gene therapy. Thank you so much to everyone for joining and to our public speakers in session 1 for your presentation and remarks. My name is Dr. Helka Peredo Pinto. I am a physician in OTP's Office of Clinical Evaluation. I am also your moderator for session. We will focus on gene therapy and products involving gene editing techniques. Before we begin, I want to share some reminders with our public speakers. During this event, all microphones for the general audience have been muted. Because this is a listening meeting, we will not have Q&A portion for the audience, but we do appreciate your comments and encourage you to go to the docket if you would like to share specific feedback on these topics. As speakers, we appreciate your interest in participating in today. In today's meeting. Speakers, we appreciate your interest in participating in today's meeting. We will introduce each speaker so that you can begin your presentation. When it's your turn to speak, you will be asked to unmute yourself. Once your presentation is done, you'll be asked to go back on mute and allow the speaker to present. Our speakers in session 2 should see their microphone and camera access if you are a session 2 speaker and you don't have a microphone or camera access, please let us know in the chat box. For those of you who submitted the slides, your slide presentation has been added to this slide deck. When speaking, please let us know when to advance the slides. For those of you who submitted your slide presentations, as I said before, let us know when to advance those slides. Before beginning your presentations, please state your name and your affiliation to ensure transparency as this listening meeting. We encourage you to advise the audience of any financial relationship that you may have with any firm, group, company, or product at the start of your presentation. If you don't have any financial relationships as such, you can simply make a statement to that effect. Please keep the time limit in mind when speaking. Each speaker will have 5 minutes. If you run out of time, we encourage you to submit remaining comments to the docket. And once again, thank you to our public speakers for taking the time to share our perspective today. Next slide, please. As with session 1, I would like to take a moment to introduce the FDA OTP subject matter experts who are serving on today's panel for session 2. These panelists will be taking notes during the session 2 presentations, and at the end of the session, the panelists may have questions for the public speakers. Dr. Young-Woon Choi, Bioinformatics Reviewer in the Office of Cellular Therapy and Human Tissue. Dr. Estella Lee, CMC Reviewer in the Office of Gene Therapy. Dr. Mondana McKean, Lead Biologist in the Office of Pharmacology and Toxicology. And Dr. Ashley Munchel, Clinical Reviewer in the Office of Clinical Evaluation. I want to thank you, our FDA OTP panelists, for your time today. Next slide, please. And with that, we will move on to right into section 2. Our first speaker for session 2 is Mr. Basel Asaf on behalf of Attentive Science. Mr. Asaf, are you ready to present? If so, please unmute your microphone, begin speaking. Your microphone should be open.
Thank you so much for this opportunity to present in this listening session, and thanks for the FDA for setting up such a vital and important initiative to improve communication and improve on the drug development of gene therapy and gene editing modalities. My name is Basel Asaf. I'm a VP of Toxicology and Pathology at Attentive Science. My financial interests, I have stocks from my previous employers at Sanofi and Pfizer. Next slide, please. The opinions expressed in this presentation are those of mine and not my current or previous employers. Next slide. I wanted to start to emphasize the value of gene therapy and how it holds and currently and in the future great promise for patients, the healthcare system, and the pharmaceutical industry. There are several FDA-approved products with a demonstrable value to the patient and the healthcare system, and we are very familiar with. It's a well-established technology, whether you're using an AAV, lipid nanoparticle, or lentivirus, and it continues to be further and further refined. There's a strong scientific foundation for it and a plethora of data accessible in the public domain that helps current and future drug developers. Although there has been some unfortunate events, we learned a lot from those, and this is natural progress of science that we learn from these lessons. And the good thing is we have a lot of mitigation strategies handy and available to address some of these issues. Ultimately, there are huge future opportunities from these modalities, including maximizing on existing data and establishing platform approaches that ultimately shall reduce resources and accelerate product development and providing patients with transformative medicines at affordable cost. Next slide. I'm making my presentation into three different proposals. The first one is leveraging existing knowledge of AAV to align on best practices. At this current stage, we have a lot of knowledge in the literature and internal knowledge within the institutions on AAV biology, drug development, and performance in the clinic. There's a large body of non-clinical biodistribution data that is tested under various conditions, such as different routes of administration, dose levels, and so on. And many of those are in the public domain. So just as an example for AAV2, AAV8, and AAV9. And those can strongly be leveraged to eliminate or reduce biodistribution needs, especially for well-established capsids. We know from experience, as well as in the published domain, that non-clinical toxicology studies longer than 3 months rarely identify new clinically meaningful findings. Now, it doesn't mean that longer studies are not needed, but the identification of novel findings have been all confined within the first 3 months, and longer studies typically are used to assess reversibility, recovery, or lack of progression of some findings. And this understanding may allow us to limit the IND-enabling studies to 3 months duration unless scientifically warranted to run longer studies. And this would help sponsors and drug developers to predict what is needed in their planning for cost and time for their product development. Interim necropsy are very valuable in general. The genesis of this comes from small molecules or biologics involved in repeat dosing, in which you could initiate first in human with shorter-term studies and then subsequently plan for chronic studies. However, gene therapy is intended from the beginning to be a durable, lifelong product. And consequently, the value of interim necropsy, although useful, it is not critical compared to the terminal one, which is typically the longer-term endpoint in the study. And the proposal here is to leverage some of the non-GLP studies that are assessed early on in the program to supplement for these short-term assessments and potentially eliminate interim necropsy time points from the GLP toxicity study, albeit reasonable comparability is there between any research-grade material versus GLP and GMP material. And finally, sponsors, whether they do it individually or through consortia, might be willing to share some of that data if it's masked or if it's put behind membership wall, essentially, to limit access or at least monitor access. And this is not unheard of. Even in the scientific literature, unfortunately, we often know that negative data is rarely published, and in drug development, failed products are rarely shared and published. Consequently, being able to share all of that information for products that even those that will not make it forward will be very, very valuable for the field. And the proposal ultimately is to improve on aligning on best practices to streamline non-clinical development of AAV gene therapy products. Next slide. The second proposal here is further refining on the biodistribution need. As I mentioned in the previous slide, large body of literature and FDA-approved products in the public domain are available that can potentially eliminate or at least reduce the need for some of the biodistribution endpoints. Animal biodistribution data have not added value for non-target tissues that do not exhibit evidence of toxicity. And one could propose potentially collecting and storing extensive tissue lists, but not require it for phase 1 clinical trial, and only trigger it if toxicity is seen, for example, in the kidney. We rarely see toxicity of AAV in the kidney, yet we always analyze it. And we collect the data and we just put it on the shelf without using it truly. Third, translatability of animal to human biodistribution data remains unknown, which is totally understandable because it's not feasible to collect extensive tissue lists the same way we can do in animals. Consequently, establishing translatable models will provide a great value to the field. And finally, just as proposed previously.
The next speaker. I'm sorry. So unfortunately, your time is up. I have to move to the next speaker. However, we encourage you to please add a reminder of your presentation to the docket. Thank you very much.
Thank you.
With that, we'll go to the next speaker, who is A.B. Edgeman on behalf of Tesara Therapeutics.
Good afternoon. Thank you to the FDA for organizing this listening meeting today, which I believe will ultimately benefit patients. I'm Anne-Virginie Eggeman. I'm Chief Regulatory Officer at Tesara Therapeutics. I'm on the advisory board of Rarity Public Benefit Corporation, and I'm also the industry representative of the FDA Cellular Tissue and Gene Therapy Advisory Committee. My presentation will focus on how to leverage knowledge to facilitate both the development and the review of cell and gene therapy products. In addition, I will refer to cell and gene therapies as CGT for brevity. Next slide. The optimal way to leverage prior knowledge to facilitate the development of CGT products, in my view, is to use a platform approach very early in development, starting at the development candidate nomination stage. First, we recommend the agency creates and/or facilitates early platform meetings where the development of several products leveraging the same prior knowledge could be discussed. It may be helpful for the FDA to amend internal SOPs to allow efficient organization of such meetings. The prior knowledge discussed at such meeting could, in the future, form the basis of a platform technology designation after the first product leveraging this prior knowledge is approved. Second, we recommend the agency either amend existing CGT guidelines or publish a new platform guidance specific to CGT. These amendments should give examples of acceptable situations where prior knowledge can be leveraged. These examples could include, as mentioned this morning, shortened in vivo toxicology study or toxicology conducted in only one species, or if enough data are available, for example, no in vivo toxicology study for future products. Other speakers also spoke about not repeating biodistribution studies for genome editing products using the same lipid delivery vehicle. We agree this is also a good example. Importantly, these examples should include CMC, non-clinical, and clinical development, not just one discipline. Next slide. Here are the responses to FDA-specific questions. For the first questions, for LNP in vivo genome editing products, using the same lipid delivery vehicle should allow leveraging of existing acute toxicology and PK data. Using the same components across several products should allow leveraging CMC methods and data for analytical testing. Developing several similar products, each for a distinct disease-causing variant in the same gene for the same disease, should allow extrapolation of CMC and non-clinical data across products, and should allow conducting only one umbrella clinical trial with all products. For the second question, leveraging manufacturing data for INDs and BLAs from CDMOs should be feasible. Allowing the use of a drug master file in BLAs will be key for this solution, which we realize will require modifying existing regulations. For the third question, data and information can be effectively integrated across multiple disciplines by having early platform meetings, which we just discussed in the prior slide. Continuing RMAT post-designation cross-functional forward-looking meetings will also help. I also agree with prior speakers that integrating such cross-functional platform considerations in interact and/or pre-IND meetings could be helpful. For the last question, the key to facilitate data and information sharing will be both the creation of a plasma master file, which I will discuss on the next slide, as well as continuing open dialogue at workshops or listening sessions like today. Next slide. To improve the review of CGT dossiers, we recommend two things. First, the creation of a cloud-based review system. This would avoid using emails, which can be lost. This would allow for both sponsors and FDA to see the same information and to exchange efficient correspondence. Our PMs and/or reviewers could use this information to answer sponsors' questions gating for development, and vice versa for the agency to ask questions gating for finalization of review. This should overall help expedite the regulatory process. The second recommendation is to allow sponsors to create a platform master file for both INDs and BLAs. Several products could refer to this PMF without having to redo experiments, and reviewers would not have to re-review this information referred to in the PMF. Sponsors, though, should be very clear in each of their INDs and BLAs about the prior knowledge data that they are leveraging from the PMF and the time it will save in development not to redo these studies described in the PMF. Thank you for the opportunity to speak today.
Great. Thank you very much. Next speaker will be Michael Hashisawa on behalf of PCHIP Corporation. Mr. Hashisawa, if you are here, please type that you are here. In the meantime, we can call next speaker, which is Chris Saha on behalf of the University of Wisconsin-Madison. Please go ahead, Mr. Saha.
Hi, sorry, that's actually my presentation. If you'd like me to go now.
Yeah, sorry, that's fine. Oh, yeah, Mr. Limicki from Alliance for Regenerative Medicine is next. Thank you very much.
Thank you. My name is Michael Lemicki. I'm the Senior Vice President at the Alliance for Regenerative Medicine, or ARM for short. I want to thank FDA and OTP for organizing this important meeting and allowing me to speak on behalf of our members. We are a not-for-profit advocacy group with over 400 members, cell and gene therapy developers, supporting companies, academic centers, and patient organizations. I'm going to speak today about leveraging data via platform principles in gene editing. Next slide, please. The concepts that I will speak about today are the product of an ARM meeting called the Scientific Exchange. The Scientific Exchange is a forum for open information exchange. We convened this meeting with the support of ISCT and Danaher to share best practices for employing platform principles in the development of gene therapies for rare disease. Gene editing is uniquely able to address many minor genetic disorders, including rare diseases with multiple genetic variants. Currently, the regulations do not formally identify how platform principles may be used to streamline development. We invited academic KOLs and developers to present case studies, both real and hypothetical, about how platform principles may be applied in the development of gene editing therapeutics. FDA experts and senior leadership were present to listen and comment on the merits of the case studies. EMA and MHRA also participated in listen-only mode. During the meeting, we identified several key themes, which are shown on the right side of this slide, mainly by holding the vector or the delivery vehicle constant using a platform approach. This facilitates streamlining biodistribution and tox testing. Consistent manufacturing steps in a platform can be used to streamline validation and release testing. I'll speak in a subsequent slide a little more about the importance of benefit-risk considerations in applying GMP. And last but not least, innovative umbrella clinical trial designs can be used, as the last speaker referenced, in situations where you're studying a disease with multiple genetic variants. The participants predicted that these principles could lead to 5-fold material efficiency gains and decrease the time required to dose the first patient to as little as 6 months, which we have subsequently seen to become a reality in the well-known case of baby KJ. Next slide, please. These are some additional principles that we have already shared with FDA leadership, but I'll briefly cover them today. Gene editing uses standardized, repeatable components such as delivery vehicles, gene editing enzymes, and manufacturing processes applied across multiple therapies. Nevertheless, they face long and costly development timelines, as they are often treated, each product is treated as a bespoke therapy, even when it shares common elements with other products in a platform. We recognize the importance of the platform technology designation. However, the limitation that this platform designation be based on an approved product is a significant barrier for gene therapies targeting rare disease. The agency has announced plans to issue specific guidance on platform applications to gene editing, and we applaud that, along with the plans to issue guidance related to today's topic, and we encourage the agency to prioritize this. Solutions include defining the criteria for a gene editing platform in terms of the vector, the editor, and modular guide RNAs, for example, enabling cross-referencing of prior INDs for CMC, preclinical, and clinical modules, developing evidentiary guidelines for expanding platforms across new indications, and allowing master protocol approaches with shared clinical infrastructure and long-term follow-up. Next slide. On my last slide, I'll talk about some CMC-specific issues. We term this risk-appropriate CMC. The situation is that CMC requirements for gene therapies and gene editing therapies for rare diseases are equivalent and sometimes more stringent than CMC requirements for larger indications. This creates a significant burden. The level of GMP required at early phases leads to very high costs for small batches of raw materials. A good example is a GMP run of guide RNA, which can easily run into 7 figures. Validation and release testing consume a significant amount of small batches of drug product. Solutions to these problems, we believe purity requirements for guide RNA should be revisited in situations where the benefit to patients exceeds the risks associated with off-target editing. The size of the patient population should be taken into account when determining the sample size for CMC test requirements. As many speakers today have spoken about already, apply platform principles and prior knowledge to streamline testing, especially comparability test requirements. Eliminate the requirement for multiple PPQ runs in situations where batches are produced with low frequency. And finally, allow certain CMC requirements to be met in a post-market setting. Most of these solutions have already been applied at one time or another on a case-by-case basis. We feel they're non-controversial, and we would recommend that the agency apply them more consistently and provide some additional guidance on how they may be applied. And with that, I will close and thank you again. And I invite any questions or additional discussion at the end of the session.
Great. Thank you very, very much. Our next speaker is Chris Saha on behalf of the University of Wisconsin-Madison.
Thank you, Helga, and thank you to the organizers for the opportunity to speak today. Good afternoon. I'm Chris Saha from the University of Wisconsin-Madison. I am on the advisory boards of Janssen Biotech and Bharat Biotech. Today, I will describe my efforts within a larger group led by academia to advance gene editing drug development. Next slide, please. My lab within the NIH Somatic Cell Genome Editing Consortium is developing non-viral gene editing platforms for inherited retinal disorders. Our lead program for a form of pediatric blindness uses a lipid nanoparticle to deliver an adenine base editor and a guide RNA targeting a pathogenic mutation. Within patient-derived cells, we have demonstrated correction of the KCNJ13 mutation and restoration of resulting KIR7.1 channel function. However, the path to a first-in-human study presents many challenges. 3 challenges among many that we're tackling within our program are listed here. First, for CMC, what is the definitive data package required for the novel ionizable lipids in our LNP formulation? Which of these are considered novel excipients, for example? Second, for safety, for every new guide RNA targeting a different mutation, how do we quickly conduct a comprehensive genome-wide off-target analysis using the latest nomination methods? And third, for our clinical plan, what constitutes a sufficient natural history comparator and long-term follow-up protocol for an ultra-rare pediatric population? These challenges are not unique to us, but these types of questions, as we have heard from others, can be rate limiting for many in this field. Next slide, please. But despite these challenges, as was just alluded to, we have a powerful blueprint for what's possible. The landmark Baby KJ case, led by a team in the NIH SCGE consortium, used a similar platform, an adenine base editor, delivered via a lipid nanoparticle for an in vivo application. This heroic multi-institutional effort generated the necessary preclinical data package from off-target nomination to IND-enabling toxicology studies in dialogue with the FDA in record time. This N of 1 success was undoubtedly a scientific triumph, but it also serves as a warning. This bespoke, resource-intensive model is not a scalable solution for the thousands of other rare diseases. Next slide, please. To move from the N of 1 successes to an N of many platform, we propose the formation of a broader consortium, provisionally named here an on-demand genomic medicine consortium. The mission of this group would be to build a public library of prior knowledge that could be leveraged across programs. This would include data packages on the toxicology of common LNP components, for example, standardized potency assays, and validated off-target analysis pipelines. By linking non-clinical data to clinical outcomes, potentially with AI and other machine learning techniques, this consortium can create a framework for platform designations, enabling the cross-referencing of data to significantly de-risk and accelerate IND submissions. We think this is possible for several programs within the NIH SCGE and more as well. And within the SCGE, we are looking to release some of the data that we're generating along with best practices to move this type of concept forward. Next slide, please. Next slide, please. So this really brings me to a critical alignment with the agency's goals. The FDA's new Rare Disease Evidence Principles program is really a perfect catalyst for this model. This consortium that I just described, involving academic medical centers, would be the ideal engine to generate the robust confirmatory evidence that this RDEP concept calls for, from strong mechanistic data derived from standardized assays to shared non-clinical models that can strengthen the evidence base for a single, well-confirmed, predictable path to licensure. This is how we solve the recurring technical bottlenecks for CMC safety clinical development and turn the heroic success of a one-off cure into predictable pipeline of genomic medicines. Thank you again for the opportunity to share our perspective.
Great. Thank you very, very much to all our public speakers. At this time, if our FDA OTP's panelists will have any questions to follow up, maybe we can start our questioning session in this part. Let's see. All right. Any questions from OTP? I think Estela, Dr. Lee, do you have any questions for the panelists?
Yes, thank you. Yep, thanks. Yeah, I do have a quick follow-up questions for two speakers. So hi, this is Stella Lee from OTP in Office of Gene Therapy. My first question is for AV Egimen regarding, and first of all, thank you all for great talks here. It's related to your comment about the platform master file. My question is, how do you envision the BLA applicants to be able to assess and share data in the proposed master file platform to allow them to have a full understanding of their information supporting their products? Could you please elaborate a little bit more on that comment, please?
Sure. Thank you for your question. The way I envision this is that the platform master file would be submitted by the sponsor, and I presume it could be in the shape of a BLA itself in some ways and include the platform information, either CMC, non-clinical, or clinical, that would then be leveraged in several INDs and or several BLAs over time. And this platform master file could be updated over time as well. This would require, obviously, collaboration with the agency. Does that answer your question?
Sounds good. Thank you.
Thank you.
I have another question for a different speaker from Michael Lee Mikey. Sorry if I'm not pronouncing your last name correctly.
Okay.
It was regarding your comment about revisiting purity requirements. Can you please elaborate how leveraging data from different files could inform reassessment of the purity, the guide RNA purity requirements?
Yeah,
in the context of leveraging data from different files.
Yeah, more specifically, and I'll also refer you to an example from our paper on cytotherapy, which summarizes the scientific exchange, where it was Danaher studied guide RNAs under multiple manufacturing conditions at different purity levels, and they were able to demonstrate in this example that editing efficiency plateaued at about 70% purity. So I think that type of testing can be leveraged, and also testing for guide RNAs that identifies the impurity profile. In other words, not all impurities are created equal. So, we believe these types of things should be taken into consideration with a platform approach to allow for, again, what we call a risk-appropriate GMP approach to guide RNA production, and potentially this could be used for other critical raw materials as well. And I'm happy to send you that paper if you'd like.
Yep. Thank you for asking, just answering my questions. Yep. Thank you. Sounds good.
Great. Any more questions from our OTP panelists? I think Dr. McCann.
Yes, hi, this is Mandana McCann from Pharmacology Toxicology. We have a clarifying question for Dr. Asaf. Could you please clarify or provide examples of what you're proposing to include in the reduced biodistribution to de-risk novel capsids?
So one example is, depending on what is the nature of the novel capsid and if it's derived from an already existing well-characterized capsid like AAV9, for example, because typically that modification of the capsid is intended to improve on the on-target rather than significantly change the off-target. Another example is to reduce the number of tissues, particularly for certain routes of administration like intraparenchymal injection or intraocular injection. So these are two examples, for example, for novel capsid or, already existing capsids. Yeah.
Okay, thank you for the clarification. We have another question for Dr. Saha. Could you please clarify what the FDA's role in the consortium that you proposed would be?
I think the RDEP pathway is really interesting. And we haven't really sketched that out, firmly. I think we see this as an opportunity for us to promote, leverage the prior knowledge that might be in the agency and communicate that to sponsors. But, this is part of some of the exchanges that the SCG has had over the last several years that led up to some of the programs that are advancing, including the one that we have been advancing in the retina, that build upon one another. So, I think exactly how that interaction would occur is something that we haven't, I would say, entirely sketched out, but I think there's some interesting models of success in the SCGE's interaction with FDA that give us some inspiration for that.
Okay. Thank you for the clarification. That was all my questions, Halka.
Great, thank you. So seems no more questions from the OTP no? Okay, great, thank you. So I would like to thank all our FDA OTP panelists and to all of our public speakers in session 2 for your presentation and remarks. At this time, we will take a short break and resume the public meeting in 15 minutes for our last and final session. For our public speakers in session 3, we will be giving you camera and microphone access during this break. So we ask you to please stay close by. Thank you all, and we will be back in 15 minutes.
Great. Welcome back to our public listening meeting on leveraging knowledge for facilitating the development and review of cell and gene therapies. I know that was a short break, but we're glad everyone could rejoin here for our last session. I want to take a moment to thank our public speakers in sessions 1 and 2 for sharing their perspectives on this important topic. And now we'll move into our final session for today's meeting, which will focus on cell therapy products. My name is Melanie Ego. I'm a division director in OTP's Office of Cellular Therapy and Human Tissue. I'm also your moderator for this afternoon session. Before we begin, I want to share some final reminders, repeat of earlier reminders, but about the virtual listening meeting process. During this event, all microphones for the general audience have been muted. Because this is a listening meeting, we will not have a Q&A portion of for the audience, but we do appreciate your comments and encourage you to go to the docket if you'd like to share specific feedback on these topics. We will put the docket number in the chat. Speakers, we greatly appreciate your interest in participating in today's meeting. We will introduce each speaker so that you can begin your presentation. When it is your turn to speak, you will be asked to unmute yourself. Once your presentation is done, you will be asked to go back on mute and allow the next speaker to present. Our speakers in session 3 should see their microphone and camera access. If you are a session 3 speaker and you do not have microphone or camera access at the moment, please let us know in the chat box. For those of you who submitted slides, your slide presentations have been added to the slide deck. When speaking, please let us know when to advance the slides. Before beginning your presentation, please state your name and your affiliation to ensure transparency at this listening meeting. We encourage you to advise the audience of any financial relationship that you may have with any firm, group, company, or product at the start of your presentation. If you do not have a financial relationship as such, you can simply make a statement to that effect. Please keep the time limit in mind when speaking. Each speaker will have 5 minutes, and you will notice when I come online, my video comes online, that you're nearing your 5 minutes. If you run out of time, we encourage you to submit remaining comments to the docket. Once again, thank you to our public speakers for taking the time to share your perspectives today. So as with our previous sessions, I'd like to take a moment to introduce the FDA OTP subject matter experts who are serving on today's panel for session 3. Dr. Saravana Karambayaram, CMC reviewer in the Office of Cellular Therapies and Human Tissues. Dr. Lauren Kakai, CMC reviewer in the Office of Cellular Therapies and Human Tissue. Dr. Kavita Natarajan, clinical reviewer in the Office of Clinical Evaluation. Dr. Sashi Tomar, pharmacology toxicology reviewer in the Office of Pharmacology and Toxicology. At the end of the session, the panelists may have clarifying or follow-up questions for our public speakers. We ask that our public speakers in session 3 stay on for the entirety of the session in case the panelists have a question for you. Hey, next slide, please. And with that, we will move right into session 3. Our first speaker for session 3 is Ms. Deborah Webster. Ms. Webster, are you ready to present?
Yes, I am.
Please unmute. Okay, great. Let's move right along.
So are you presenting my slides? Yes.
Yes, you can start.
Are you, but I don't see, are you presenting my slides or am I moving them?
Your slides are being presented. It's on your
okay, I see it now. Thank you. Thank you very much for this opportunity to speak with you. I'm Deborah Webster. I'm with SSI Strategies. I have no financial interest to disclose. My company works on supporting companies in medical and regulatory affairs with respect to developing advanced therapies. So next slide, please. So Oops, next slide, or do I advance it? Okay, so anyhow, thank you, Ed. I'm going to talk a little bit about the current state of tumorigenicity assessments in cell and gene therapy, cell therapies in particularly, and how well do these reliably assess risk. First of all, mouse models, or especially immunodeficient ones, have significant limitations when used in human studies. They lack the critical components of the human immune system that are essential for tumor suppression, and this can skew results, exaggerating tumor formation due to no immune clearance or missing immune-dependent mechanisms. The mouse microenvironment is fundamentally different from humans, which can affect how how tumors develop and respond to how the cells respond in that environment. Fourth, the background pathology of these strains may complicate interpretation, as spontaneous lesions or tumors may be mistaken for experimental outcomes. And thirdly, scaling issues matter. The number of cells injected into the animal and the site of injection and the mouse's size and lifespan can lead to exposures that don't translate to human biology, potentially producing misleading results, in particular with respect of intended cell engraftment being mistaken for tumor formation in a blinded histopathology review. The use of homologous therapy products are also difficult. These do not overcome the challenges and are hampered by complexity of cellular therapy, manufacturing variability, and comparability challenges. Given these limitations, there is a need to incorporate alternative assays to improve translational relevance. Next slide. A quick ChatGPT review of the guidance shows that FDA has about 5 separate guidance discussing in which tumorigenicity is discussed. Notably, in the 2013 FDA guidance on non-clinical assessment of cell and gene therapies, it's currently stated that there are no scientific consensus regarding the ability of current animal models to predict tumorigenicity. So we think that as over the course of my career in advising companies, what I've seen is a shift in FDA's experience or in thinking around tumorigenicity as they've gained more experience with cell therapy. And this seems to be evolving from a requirement for animal studies as an absolute to a more risk-based approach, including in silico, in vitro, and in vivo assessments. And an example of this in particular would be for CAR T therapies, where 2-year carcinogenicity studies have been basically replaced with integrated assessments of vector integration profiling, and then long-term follow-up in patients to monitor therapy-related malignancies. So recently, it was announced that FDA had announced a recent labeling change for Eli-cel to reflect increased hematological malignancies. And if you go and look at the summary basis of approval for this, you'll notice that non-clinical studies of the in vivo studies conducted to support Eli-cel showed no vector-related tumors observed in the animals. So again, pointing to, is this the most relevant translatable method of assessing risk of oncogenic transformation of transplanted cells or unexpected proliferation or immortalization events? We would like to propose a more bioinformatic approach, analysis of gene expression and genomic sequence in the final cell product, and in silico analysis showing no activation of oncogenes or telomerase or reactivation of pluripotency genes, and demonstration of a normal karyotype as being supportive that the cells are not predisposed to tumor formation and potentially obviate the need for an in vivo assessment of tumorigenicity. We'd like to point out that, or we'd like to make a recommendation that based on the studies that have been submitted to FDA by sponsors, that if it's possible for FDA to do a deep dive into this data using their AI capabilities, and perhaps driven by some machine learning or predictive models that can incorporate aspects associated with how we would predict oncogenicity, to come up with a more risk-based approach that encompasses non-clinical CMC tumorigenicity assessments in the context of the clinical indication. And perhaps this deep dive into their past experience with these studies in the context of these products, be able to issue a consolidated tier-based guidance approach that could lead to a more translationally relevant risk assessment that could be incorporated into the NAMS roadmap. So I thank you again very much for this opportunity to chat with you and present this opinion.
Thank you, Dr. Webster. Moving on to Jeremy Ewald. Are you here? Jeremy Ewald, can you please turn your camera and unmute if you're here? Maybe we can move on to our next speaker and come back to Jeremy Ewald. Yeah, I'm not seeing Jeremy Ewald coming on. So let's move on to Dr. Lisa Flood. On behalf of Disease-agnostic, can you please get on camera and present? Thank you.
Hello, I appreciate the time today, so thank you very much. Let me just clarify, I'm not a doctor. But I am, my name is Lisa Flood. I'm the Senior Director of Regulatory and Clinical Operations for Disease-agnostic. I have no financial conflicts other than I am the employee of Disease-agnostic. Next slide, please. So just to say up front, Disease-agnostic is a small company based in Salt Lake City, Utah, that has a allogeneic cell therapy for injectable cell therapy for degenerative disc disease. And I thought it was interesting because there are a lot of products that are intradiscal injections out right now. Some are approved, some are not. And I thought that I would speak on sponsored data sharing with regard to what we've been experiencing or just give some suggestions. For data sharing, it's a little difficult because a lot of these new innovations are novel, and it's hard to pull data when you have different cells, but I do think that there could be a benefit to it. Some of the key factors, though, are the competitive landscape. It is a very competitive area right now. There are some benefits to data pooling. There is somewhat of a corporate or proprietary risk, and then it also depends on clinical stage of development. Next slide. What types of data are sponsors willing to share? So first and foremost, safety data. I think that long-term post-market and pooled adverse events are very important. And even efficacy trends, any durable effects that might be seen, endpoints in accelerated approval, and then process learnings, which would be trial design, comparability case studies, and the like. Having that continuity across similar trials and having that data that's pooled and shared could be beneficial. There is less willingness to share any interim data in a current ongoing clinical trial, or any proprietary primary efficacy data prior to approval or publication. Next slide. The benefits are that it could potentially accelerate regulatory review. It can improve cross-product comparability. It enables smarter trial designs, especially with regard to adaptive trial designs. And for example, sample size reassessment. It enhances early identification of risks when data is pooled. It reduces duplication across sponsors, and it supports early identification of treatment-specific risks. Next slide. Potential sources for data pooling, specifically with regard to safety, would be industry-led clinical trials, investigator-initiated clinical trials, academic and institutional clinical trials, and post-marketing registry and surveillance studies. Next slide. This is sort of my little way of seeing how all of these different areas of research could pool the data into an FDA-led hub that could be potentially beneficial to all of the parties involved. And I see this as a framework for a collaboration across stakeholders. I think this model also could be applicable to just about any disease indication or platform. Next slide, please. My recommendations, and this is where I get to a little bit of the meat of it, is that if we're going to pool data, we should utilize confidential data that has been de-identified and is anonymous, so that there's not that fear of any competitive risk, and that it would be appropriate and similar comparative data. Another suggestion would be to broaden surrogate endpoints for acceptance of cell and gene therapies across trials. More of a use of an adaptive trial design to incentivize data pooling. And then regulatory incentives, I think, are the most important thing, which would be if the sponsor or any of the academic institutions or anybody that would be contributing to the hub were given any kind of incentives, such as extended exclusivity for sharing their data, reduced fees tied to transparent data sharing. And then one that affects us the most would be early regulatory review of primary and secondary efficacy data, which is sort of an interim analysis. So we also, I should have said, are RMAT and fast track designated. Therefore, we have the potential for an accelerated approval. However, we don't have surrogate endpoints. We don't have surrogate and intermediate endpoints. We only have primary and secondary efficacy endpoints, and then long-term follow-up after that. So one of the things that I think in data sharing and in getting early interim results would be that there would be a consideration for larger indications such as degenerative disc disease, as opposed to just rare and oncology, and that whole mindset gets expanded, and then it doesn't always have to be surrogate.
Ms. Flood, I'm sorry, but I will have to interrupt you. Unfortunately, we are out of time.
I'm sorry. And thank you very much for your time.
Please submit remainder of your presentation to the docket. Thank you. Moving on to Bambi Grilly.
Oops. Hello, can you hear me?
Yes, and your slide is started.
So I want to talk to you today about a case study that's viral specific T cells with a focus on kind of this concept of data sharing. It kind of builds off of Lisa Flood's talk, so I'm glad I'm in this position. My name is Bambi Grilly. I'm the head of regulatory affairs at Baylor College of Medicine Center for Cell and Gene Therapy. I own a consulting company that does work in the same arena for investigators and small companies, and I'm also the chief regulatory officer for the International Society for Cell and Gene Therapy. Next slide. You heard about ISCT earlier, but basically we're a group of people that have come together with a shared vision to translate cell and gene therapy into therapies that improve patients' lives worldwide. Next slide. And so again, this is just to talk about a case study. Next slide. The history of the case study is that viral infections are a significant problem post-transplant. This is due to the absence of T cells, and it led to the development of a lot of products, T cell products, over the decades following the 1990s, starting with EBV cytotoxic T lymphocytes, and kind of moving through a variety of different approaches. Development of these products has been ongoing at multiple institutions and in some companies with some variability in manufacturing. However, overall, the cell product is better defined by its composition and its activity than by its manufacturing, with the primary caveat there being that autologous and allogeneic products have to be considered separately. Next slide. In this arena, we've seen confirmed activity in multiple centers with sustained response rates of 80 to 95% in donor-specific studies and 60 to 90% in third-party VSTs. The products have a low risk profile, and this is irrespective of the manufacturing method, meaning irrespective of which center is reporting. Next slide. The primary focus or the primary objective here is actually off-the-shelf T cell therapies, where you can select the donor with optimal characteristics. The products are immediately available instead of needing to wait for manufacturing. They're cost-effective and scalable. And the primary challenge is alloreactivity, with the possibility of increased risk of GvHD, or limited persistence. Next slide. In this arena, we've seen confirmed activity in multiple centers with sustained response rates of 80 to 95% in donor-specific studies and 60 to 90% in third-party VSTs. The products have a low risk profile, and this is irrespective of the manufacturing method, meaning irrespective of which center is reporting. Next slide. Next slide. So Alavir is a company that, oops, I'm sorry. We're one too far. Sorry, can we go back? Alavir is a company that was going to take these products and try to develop them as marketed products. They did 2 phase 2 studies, one in kidney transplant patients with BK viremia and one in phase 2 for prophylaxis. Next slide. And then they did 3 phase 3 studies, one that was prophylaxis, and I want to emphasize that prophylaxis was not the primary focus of the initial studies of the product. One using BK to treat BK-induced hemorrhagic cystitis, which was also not part of the initial studies, and one for adenoviral infections. Next slide. Next slide. The phase 2 study showed encouraging safety, and we think that the failure of the phase 3 trials to meet their endpoints illustrates challenges that might include early termination limited to their ability to assess secondary outcomes, the complexity of these patient models, and also that the effectiveness of these therapies may depend on patient-specific immune factors, viral loads, and immune suppression. With that in mind, the commercial development of this product was discontinued. However, there remains an ongoing clinician desire for these therapies. Earlier this year, leaders in the field met to discuss a way forward. And one of the things that we've talked about is what will future models of care look like? Will we have academic banks looking like cord banks? Will we have banks in different countries like the US, Europe, or Australia? Next slide. And as we push this forward, not only to include VSTs after HSCT, but also to include solid organ transplant, what are the biggest challenges to making these available to patients? And I would say the number one challenge is funding and reimbursement. There's also the question of whether there's a path forward with commercialization, which would frankly require filing of a joint BLA. And are there alternatives to commercialization, which would include a robust cost recovery model that includes a method of enforcement, because that's the major stumbling block of the existing cost recovery model. Next slide. But overall, what is the biggest hurdle? The biggest hurdle is that with over 30 years of data and over 2,100 patients treated, using the existing data evaluation standards, the information remains fragmented, and we're unable to pull together a complete picture of the success of these products. Next slide. So sharing of verified data, and this is where I want to emphasize what Lisa Flood said, we could have increased rapid product development, early to data availability. We could have helped move R&D costs to the cost of manufacturing, and we could help aggregate data to support licensing. And I know my time is up, so thank you very much. I appreciate your time.
Thank you. All right, let's move to Matthew Hewitt from Charles River Laboratories. Can you please come online, unmute? Matthew Hewitt. Okay, going once, twice. All right, we'll move on and maybe hit upon you again, Matthew Hewitt, at the end. As to moving to Deepa Chand on behalf of Novartis.
Hello, can you hear and see me?
I can hear you.
Okay.
Now I can see you.
Terrific. Great. Thank you for the opportunity, and we'll jump right into it. If we could go ahead and go to the next slide, please. I am an employee and shareholder at Novartis, and this is my disclosure. Next slide, please. Today, I'd like to present to you our mission statement, and that is that we would propose a cross-functional collaboration is essential to improve post-marketing data collection methodology and data quality in a pragmatic, harmonized manner so that we can ensure patient safety, especially for CAR T therapies. Next slide. And to dive into that a little bit deeper, I'd like to discuss a bit about post-marketing safety monitoring. When we do clinical trials, we have a robust methodology for conducting our trials, and part of that includes rigorous safety monitoring through adverse event collection, laboratory monitoring, vital sign collection, and putting all of this together in a very harmonized, organized manner. When we talk about post-marketing monitoring, and this is done for every product, even after approval, and including a lot of data mining. So the methodology changes. And here, what we're now evaluating is the totality of the data in real-world use. And this includes surveillance of spontaneous reports, literature mining, social media monitoring, as well as reports received directly from health authorities. And this continues really throughout the life of the product itself. One adjuvant tool that we often use are registries, and registries can complement safety data monitoring so that we can look at specific safety outcomes, and we can do this in a prospective manner. Next slide, please. Well, when we do some of these registries, we have to keep in mind, as we do with any data that we collect, that there may be limitations. And really, when we're talking about doing registries in a post-marketing setting, we're talking about real-world use. And as we talk about this during real-world use, we have to keep the patient in mind. And when we use this patient-centric approach, then what we're doing is we're really focusing the data that we're collecting. And we have to keep in mind that when we look at this data, that the data collection might be cumbersome to patients. These are patients that have often undergone significant therapies, especially with CAR-T, where they may have experienced a lot of emotional setbacks, a lot of positive emotions as well, but also that they are trying to reintegrate into society. And we know that as they go through their care journey, that our goal is to improve quality of life. But then what we're doing is asking them to kind of revisit that illness so that we can collect data. So we need to be mindful that we don't want to be burdensome. Otherwise, what could really happen is that these patients do not provide us the information that they're looking for, and that we're looking for, and then the data collection becomes very limited. Having said that, that might be a limitation, but we also need to recognize that there might be great potential in some of this data collection. If the data is collected properly, it's synthesized and analyzed, and key messages developed, we can obtain highly useful information that can guide further safety measures for these patients. However, what we need to do is remember that we don't live in a silo. Rather, that this is another source of data for key information that we need in order to ensure patient safety. Next slide, please. So what does that mean? That means looking forward, we really should look at building things the way we want to in the future. We need a unified post-marketing safety surveillance way to look at our data. And the FDA can provide a stewardship and should provide that stewardship so that it can help us as drug developers, us as patients, us as care deliverers, collect essential data for CAR-T therapies. And the reason the FDA should be the steward of this is that the FDA has wonderful insights from various stakeholders, including the drug developers, including advocacy groups, in other words, including multiple stakeholders. And this harmonization can be done through unified registries that could be run by a neutral entity, but enable sponsors to come together and streamline that data collection for providers, for patients, and really make this a positive experience for the patient, and yet continue to advance science, which is critical in this evolving group of therapies. And with that hope, what we would like to see is a simplified, more comprehensive monitoring approach that is easier for all stakeholders, physicians, patients, regulators, and industry, so that we can enhance our scientific knowledge. Next slide, please. And with that, I would leave you with these thoughts, and thank you again for the opportunity to present.
Thank you so much, Ms. Chand. And we have Matthew Hewitt back online, so hopefully the technical glitches worked out. If you can get back online, Matthew Hewitt.
Can you hear me?
I can hear you.
Okay, that's a start.
Yes.
So I guess just if I could get my slides. Perfect. Yeah, hi everybody, Matt Hewitt. I'm the Chief Technical Officer for the manufacturing business division at Charles River, which encompasses several businesses for us. But today I'll be specifically talking about some of the work that we're doing in advanced therapeutics, as well as the ways in which we believe we can streamline advanced therapy CMC with risk-appropriate principles that don't compromise product quality or patient safety. And my only disclosure is that I do own stock from my current employer, Charles River Laboratories. Next slide, please. So I think the theme that we've heard quite a bit about, and it's one of the advantages of being later in the sessions, is that there is increased opportunities for, or we're looking for increased opportunities for data sharing in the field. And I think we need to differentiate between kind of IP, trade secrets versus common know-how, and how we create a balance, hopefully, that allows us to share different information amongst each other without compromising sensitive information. On the slide here, we're showing two examples from the BLA filings of two cell therapies, and you can see there is quite a bit of redaction here across the different methods and acceptance criteria. This potentially presents an opportunity to look at these redactions and understand, do we need all these redactions? And we're talking about redactions of CQAs rather than proprietary methods, because we certainly realize the need to protect those. But ultimately, crowdsourcing and sharing a lot of these CQAs, as well as potentially molecular test methods, could help us streamline the review process for FDA, so that we can somewhat standardize some of these, and standardization is always something we're looking to do without impinging on innovation. Next slide. Similar to that, another concept that we're looking at is, can we streamline in vivo potency? And this may be something that we can look at doing both at the clinical stage, but also potentially at the commercial stage or post-commercial stage. In the case of CAR-Ts, this is a potency assay where we know what the CAR construct is and which antigen it's supposed to target, and then we can show antigen or product specificity and the targeting of certain antigens that produces a specific immune response. In this case, many times we're looking at immune mediator secretion. And if we can compile data such that we can show that with a very high degree and level of certainty, that if we see expression of a CAR construct, that we will see engagement of that CAR with an appropriate antigen and lead to immune-mediated secretion, then the question becomes, can we streamline potency to eliminate the cell-based assay and just go with correlating expression of the CAR to potency? And that is a question that we would like to pose to the agency and understand their thinking around it. We realize that this would be a process and probably a process that will require a good bit of data to be submitted so that we could show with, again, a high level of certainty that we would see this linkage between expression and potency. Next slide. The last concept I want to talk about is purely within the manufacturing space. Right now, we're doing APS, or aseptic process simulation studies, or media fills. And with those, we're validating that an aseptic manufacturing process is successfully carried out, and that we can do so without introducing microbial contamination. Generally, this is done in an accelerated fashion where we're more or less modeling all the connection points of this process. And so this challenges processes under worst case conditions to assess multiple parts of the manufacturing process. Now, this was originally developed for processes that were run very infrequently and where teams might not be as familiar with the process on an ongoing basis, and therefore needed these processes to refresh them and make sure that they could still perform these processes in an aseptic manner. I would say that if we look at a risk-based approach here, specifically with autologous cell therapy, we are doing thousands of these processes per year, which are quite similar. And so we should look at whether we can streamline this by using existing manufacturing data of the product itself to inform us of whether or not we're doing things aseptically, and if so, could we potentially streamline or even potentially eliminate APS studies going forward in certain processes. And so this would help us streamline the manufacturing process, allow us to deliver more doses to patients while not compromise patient safety or product quality. And with that, I thank you, and we'll happy to be taking questions at the end of the session.
Thank you. Right on time. Moving on to breakthrough T1D. Marjana Marinak, if you're online.
I'm here.
Unmute. Okay, I can hear you. And slides are up.
Thank you. Good afternoon. My name is Mariana Marinak, and my disclosure is that I'm an employee of Breakthrough T1D. I'd like to thank the FDA for hosting this listening session and for the opportunity to share some perspectives. Next slide, please. Breakthrough T1D is the leading nonprofit global patient organization funding type 1 diabetes, or T1D, research. Our mission is to accelerate life-changing breakthroughs to cure, prevent, and better treat type 1 diabetes and its complications. Next slide, please. T1D is an autoimmune disease in which insulin-producing beta cells in the pancreas are destroyed, leading to lifelong dependence on exogenous insulin. All people living with type 1 diabetes require insulin to survive, but insulin is not a cure. Despite advances in care and technology, the unmet needs are still significant, which is why advancing our work in cell therapies is critical to our mission. Next slide, please. I'd like to share two examples of prior knowledge and experience that leverages data from islet transplantation that can be used to facilitate future T1D cell therapy product development and review. Next slide, please. Criteria were published in 2018 to address the lack of standardized definitions for graft function and clinical success for beta cell replacement therapies, and to help bring alignment with glycemic control metrics available from continuous glucose monitoring, or CGMs, and artificial pancreas systems. These criteria were assessed after 2 years of implementation at various clinical and research settings, with refinements published in 2021 to include CGM-based metrics. Next slide, please. Here is another example of leveraging existing data coming out of the Collaborative Islet Transplant Registry, which was established to monitor islet transplantation progress and safety. C-peptide is used as a marker of islet graft function because it reflects endogenous insulin production. Data from the registry using islet-alone transplant recipients was used to evaluate the predictive value of fasting and stimulated C-peptide levels for the 7 primary outcomes that are listed in the table. Next slide, please. Leveraging this existing data and knowledge, leaders in islet transplantation research, along with Breakthrough T1D, published refined goals for beta cell replacement therapies for the good and optimal categories, with expansion of outcomes to include CGM-based metrics such as time in, below, and above range, as well as the inclusion of clinically meaningful level 2 hypoglycemia, which are blood glucose values of less than 54 milligrams per deciliter, and C-peptide thresholds. These metrics should be used in research, development, and review of future beta cell replacement therapies in type 1 diabetes. Next slide, please. I'd also like to highlight another way in which organizations like Breakthrough T1D can help leverage existing knowledge and experience. The Breakthrough T1D Beta Cell Replacement Consortium brings together researchers and key stakeholders across multiple disciplines to address the main challenges around cell source, cell protection, and survival. Projects funded span non-clinical and clinical studies, as well as cell source manufacturing and scalability. And several funded projects from the consortium's work are in ongoing clinical trials, and several are moving forward toward first-in-human studies. Aggregated data sets of sufficient quality and standardization from consortiums such as this one could be leveraged to train AI and machine learning tools to improve preclinical testing and manufacturing predictability. Next slide, please. We've also recently published a roadmap that outlines how future generations of cell therapies and T1D will evolve. As these therapies evolve, so must the clinical trial design considerations, particularly the expansion of people with T1D who are eligible to participate, as the risks and unmet needs of our community should not be limited to the experience of severe hypoglycemic events. These are important considerations for sponsors and regulators. Data from patient preference studies, along with evidence on the impact of these therapies on how patients feel and function, should also be utilized to ensure appropriate risk-benefit assessments. We are also leveraging publicly available and existing data to develop 2 additional roadmaps covering non-clinical studies and manufacturing considerations to facilitate the development of next-generation cell therapies in type 1 diabetes as outlined in this roadmap. Next slide, please. Breakthrough T1D can be a partner and facilitator to support efforts leveraging existing data and knowledge to advance the development and review of cell therapies. The clinical outcomes assessed from islet transplantation registries and studies provide standards by which to assess outcomes across interventions and can also be used as endpoints to assess the safety and efficacy of the next generation of cell therapies in type 1 diabetes. Identifying evidence needs and standards for data collection and sharing will be critical to facilitating product development and review, and we look forward to partnering further with the FDA and stakeholders to advance cell therapies for type 1 diabetes. Thank you.
Thank you, Dr. Marinak. Moving to Dr. Laura Nicholson on behalf of Humacyte.
Hi, thank you. I'm probably the only person who doesn't have slides this afternoon, but my name is Laura Nicholson. I'm the CEO and founder of Humacyte, and thank you very much for the opportunity to speak here today. With regard to what types of data might be best shared to facilitate development and then review of cell and gene therapy products, it's our opinion that the tools now exist to really derive a much better proteomic and lipidomic understanding of the serum components that are used in almost all of these manufacturing processes. For many processes, serum remains a black box, and it can lead to important variability in outcomes and potency and survival and differentiation state of cells. But we believe that with the onset of really advanced and powerful proteomic and lipidomic techniques, that it should become possible to devise profiles for human and fetal bovine serum that might be most effective for one cell type or another. And so I think that between companies, I think leveraging this type of information could be very helpful and could be without sacrificing too much in terms of specific knowledge. I think this would help really advance the development and speed the development of many cell therapies. As we all know, for cell and gene therapies, unlike small molecules and pharmaceuticals, each cell and gene therapy is very expensive to produce, meaning that very large clinical trials that might look for small clinical effects are really not feasible. Here are some ideas that I would propose that could accelerate the clinical development of safe and effective cell and gene therapies. First of all, I would recommend that use of an adaptive trial design or a phase 1-2-3 trial design that allows early stage clinical trials to move seamlessly into later stage clinical trials using a pre-specified and pre-agreed format between the sponsor and the agency, I think could be very powerful, because leveraging all of the data that's obtained from the entire clinical experience from phase 1 onward really increases the power of our understanding of these new therapeutics. In addition, for many patients who are receiving cell and gene therapy trials, the ethically performing a randomized trial or a placebo-controlled trial can be difficult. And for these obvious reasons, a use of historic published data and or real-world data and registries can really provide valuable benchmarks to evaluate both the safety and the efficacy of investigative cell and gene therapies. Prospective agreement between the agency and the sponsor on the real-world databases or the published comparators is really critical to move such clinical development programs forward. What has been our experience, and certainly the experience of some other folks in the cell and gene therapy space, is that for these highly specialized cell and gene therapies that are under development, if there are frequent changes in the staffing in the preclinical or in the clinical review teams at the agency, this can make achieving consensus between the sponsor and the agency. It can make achieving this consensus difficult to attain, and also difficult to retain as staff members change. I would also say that with cell and gene therapies, many of us have found that as we, despite the best efforts at designing prospectively a very thoughtful clinical trial, that it's not uncommon to have subsets of information or subsets of patients either benefit particularly well or particularly poorly from the therapy under question. And for these highly novel therapies, we think it would be advantageous to be able to have open dialogue with the clinical reviewers if there's unexpected subsets of patients who tend to do particularly well or particularly poorly, because we think this will advance clinical care overall. And those are all my comments.
Thank you. Thank you so much for sharing your thoughts. And we'll move to Bojang Kosi Shabangu. Sorry, I apologize. If you're here, can you please unmute yourself? If you're not, or in case you're having audio problems, can you please type into the meeting chat to let us know that you're here? So we'll move on and see if Mr. Shabangu gets online. So moving next to Philip Vanek on behalf of the International Society for Cell and Gene Therapy.
Yeah, so thank you everybody for the opportunity to address the panel today. I'm Phil Vanek. I'm speaking in my capacity as Chief Commercialization Officer of ISCT on the topic of addressing comparability challenges in cell therapy manufacturing using machine learning and artificial intelligence technologies. I supplied my financial disclosures in writing as I'm a paid strategic and investment advisor in the life sciences through my Redline BioAdvisors consultancy. So next slide, please. You've already been introduced by Bambi and Bruce about ISCT. I will double click on the industry committee. That is an operating committee within the organization. And our mission is really to help translational scientists, clinicians, and sponsors make smart manufacturing decisions by, for example, simplifying manufacturing, removing animal-derived components where possible, focusing on CMC, defining and refining product quality attributes, directives, embracing new manufacturing technologies, building a community of experience, and of course, putting patients first. Next slide, please. So comparability, the necessary evil in our industry. This is, again, not to underscore this, but it's ensuring the quality, safety, and efficacy of a drug product through a changed manufacturing process. And our argument would be that while necessary, this introduces a large amount of friction on the product development. And on the next slide, please. And this is particularly pernicious in the area of cellular therapies. Cellular therapies are complex living drugs. The manufacturing of these therapies remain largely manual, making production costly, time-consuming, and obviously prone to human error. They're often we have limited patient materials to work with, so making comparability studies particularly challenging, and also the use of untreated or materials from normal patients may not adequately represent changes introduced by a manufacturing change. Supply chain risks are introduced by limited suppliers of critical raw materials. The administrative costs of managing those is also a challenge. There's quite a bit of processes, the product thinking in our industry, which just makes kind of this institutionalized resistance to introduce innovative changes. And many of the materials that we think could be interchangeable are sort of implicated through biological variability, making an assessment of that change particularly difficult. And in fact, potency assays, the gold standard for monitoring a therapeutic during development and sort of informing any changes, are particularly difficult to develop and often harder to qualify and validate. Next slide, please. So I won't dwell on this, but obviously the friction introduces a cost of comparability, and summarized in this BPI article from 2024, the last part, these changes and required extended studies, these expansions increase costs, create delay timelines, and make commercializing cell and gene therapies potentially fiscally unsustainable. So next slide, please. So we believe that machine learning and AI and some of these as applied to comparability is a particularly interesting area to focus our attention. We applaud the FDA's already being proactive in this space, presenting draft and other guidances around comparability, as well as the use of artificial intelligence. Next slide, please. And in the industry committee that I mentioned in the introductory slide, the group has put forth a guidance from ISCT about the applications of artificial intelligence and machine learning and how it can be applied in cell therapy. I point to the reference below for the record, and then also just talk about a couple of the key applications that were identified in this work. Machine learning for predicting stem cell donor availability, driving adaptive CAR T manufacturing, using machine learning-based prediction for CAR T efficacy as well as toxicity, using AI-powered stem cell differentiation prediction to simplify that manufacturing process, and then ultimately, potentially a digital twin of complex biological behaviors would be a useful tool for supporting manufacturing of cell therapies. Two of the key takeaways: a lack of high-quality public data sets does limit the ability. I think other speakers before just spoke about some of those. And concerted efforts to generate well-annotated large-scale data sets for modeling training will be extremely helpful and impactful in adoption of cell and gene therapies. So last slide, please. So we think that comparability is a particularly interesting application area of AI and machine learning. We have huge amounts of biological data collected over many, many years. Instrumentation and instrumentation manufacturers continue to evolve their data structuring and portability. And as commented on earlier, by combining basic science data, manufacturing data, product quality data, patient eligibility data, and patient outcome data can reveal important process changes. I think personally that AI and ML can do for cell therapy what computational fluid dynamics, CFD, has done for aerospace and automotive industries, and we urge the FDA's continued commitment to enable sponsors, investigators, and regulators to share anonymized data and to build next generation in silico biological models to power lower cost, safer, and more effective medicines. So thank you.
Thank you very much. Okay, we'll move on to Noah Scher from Inovia Therapeutics.
Hi. So it's a pleasure to be here today. Thank you for letting me have the opportunity to speak. Dr. Noah Scher, Chief Scientific Officer of Inovia Therapeutics, and as such, I'm an employee of Inovia. Next slide, please. It's a pleasure to wrap up this session. We heard today people discussing sharing of sponsor data with respect to clinical trials and post-marketing and sharing the CMC insights. Inovia is developing a CD34 stem cell-based therapy, and we also want to urge data and knowledge sharing between companies, medical institutions, CDMOs. Anything that has to do, in our mind, with the general process or analytical steps that are related to the cell therapy modality. In our case, the CD34 cells, but it could be TILs or CAR T or any other shared modality, would be extremely beneficial for the entire community. In our case, the first step is hematopoietic stem and progenitor cell mobilization and apheresis. And so shared protocols about bone marrow mobilization and SOPs that are associated with patient safety, things around bone pain or transient hypoglycemia could be shared between institutions. Apheresis parameters to help maximize yields and minimize the 9 CD34 components of these cell therapies based on hematopoietic stem cells could be shared among the community. The next step is CD34 isolation, and there's a lot of know-how and insights that are involved with this seemingly simple procedure. So any shared insights on protocols, materials, yields, instrument settings, and even differences between machines such as the Clinimax and Prodigy, and differences in using these in different patient populations would be useful. There are a lot of really interesting case studies that we've come across that we'd be happy to share, and I think if those were shared between institutions, that would be fantastic for the community. Of course, the third step is the proprietary product-related processes and analytical methods, which would be largely black boxes. In our case, it's entry of mitochondria into hematopoietic stem and progenitor cells. In another case, it might be gene related, so viral vector related, and I'm assuming those are going to be less shared between companies. And then the final phases of IV injection. How do you cryopreserve cells? What is the final formulation? And all of the release criteria, like we heard earlier from Matthew Hewitt. If we can share CMC across similar cell therapy modalities, how do we measure impurities? How do we look at potency? Some of these for sure can be shared, things like colony-forming assay information and so forth. And we think that sharing the scientific expertise with FDA guidance documents, position papers, potentially a website to share live case study updates would benefit the entire product development community and all of the stakeholders involved. And I think more than anything, it'll help us achieve better product quality, get drugs faster to patients with less duplication of effort between companies and between institutions. And the FDA stewardship would be instrumental in supporting those focused efforts around specific cell and gene therapy modalities. Thank you.
Thank you, Dr. Scher. Moving on to Jeremy Ewald from Regen Report. So Teams is working this time.
Yes, we're all good.
Awesome.
Thank you. Is it showing my slides? There we go. Hi, I'm Jeremy. I'm a journalist covering the regenerative medicine industry. I run the regenreport.com, and I'm just going to run through an interview that I just did with JSRM, that's the nonprofit that helped form the Japanese regulatory framework. The gist of it is, about 15, 20 years ago, they saw the potential for these therapies and said, hey, this doesn't fit medical device and pharmaceutical regulations. So they urged the government to make what I think most people know, the conditional approval framework. Next slide. So really broad overview. There's two tracks, one for mass manufacturing and selling these therapies on the market. That's the PMD Act for CDMOs or CMOs. And then the other track is ASRM, and that's for physicians to use on a case-by-case basis. Next slide. So PMD for the mass manufacturing, while they're doing the trials, after the early safety and efficacy has been established, they can sell these therapies on the market for only a specific amount of time while they continue to finish that trial and continue to gather data. They have to have pretty strong informed consent along the way. So they have to tell patients, hey, we don't know if this works exactly, but it looks safe and there's a little bit of data. And that's, at this point, it's actually covered by their national health insurance, which is interesting. But at any point, if endpoints are not met or there's safety or efficacy problems or something else, the government can come in and say party's over, and that's it. Next slide. Here's a list of approved regenerative medicine products that they've put through the PMD Act pathway. Next slide. And the other track, ASRM, so that's a case-by-case physician-led track. So essentially, if let's say your patient has knee osteoarthritis and you think, okay, well, maybe we can take their bone marrow out and expand mesenchymal stem cells and inject that. Maybe that will help. They can submit the clinical plan to the government and get approval to use those things. Could be pluripotent derived, could be even xenogenic or gene therapies, and they use a class-based system depending on the risk profile. And essentially, the more risky that the product is, the more difficult it is to get that approval. Next slide. Here are the classes. So class 1 is most difficult for those physicians to get. Class 3 is easiest. What's interesting is that anything allogeneic, regardless if it's expanded in vitro culture or anything, it doesn't matter. If it comes from someone else's body, that's put in the same risk profile as anything pluripotent derived and genetically modified and even xenogenic. Next slide. This has brought some interesting strengths and weaknesses from what JSRM told me. For one, rapid access to new therapies, especially things that we just don't have good answers for, traumatic brain injury, spinal cord injury, blindness, lots of things that traditional medicine doesn't have much for. Now patients have access to things that maybe could help them. It's also granted ethically bound freedom for physicians, which I think is really important because especially if a physician has that one-on-one relationship with their patient over, often decades, they know that patient better than the FDA, better than any CDMO, and they can make the best decisions for that patient. So I think that that's also really important. But it's also brought some weaknesses too. Companies do what they do, and that's skirting oversight. For instance, lyophilizing a product is no longer a cell and gene therapy, and that's just outside of the scope, and they figure out their ways. That's just how it works. And there's also a lack of efficacy surveillance. So because they don't know how well it works, because the data hasn't been collected, especially in like a third-party standardized registry, there's probably a lot of arbitrary treatments happening that maybe didn't do anything or wasn't even necessary in the first place, and that causes financial harm. And also ASRM, this physician case-by-case basis, is not covered by health insurance. So yeah, and also like I mentioned, placebo-prone and no cell viability requirements, and some of this may sound familiar to the U.S. as well. Next slide. So JSRM they're developing some solutions for those problems right now. For ASRM, for the physician track, what they're hoping to do is mandate a third-party registry that they developed. What they did was they went to the Japanese version of ALS Foundation, Orthopedic Association, and all these different domains, and they had those committees sort of form what endpoints, what sort of data they should be capturing and put into that third-party registry, because measuring Alzheimer's benefit is very different than measuring knee osteoarthritis, completely different data. So they went to every single domain and developed this pretty hefty third-party registry, and they're hoping to make sure that physicians have to use this third-party registry and make that data publicly available for the next set of patients looking at that therapy. Next slide. So what do we do? Well, you have to, I think we know USA is not Japan. We're actually quite different. So for one, we don't have public health insurance with private health insurance. We're also a little less healthy. We're also pretty ferociously capitalist, and we're pretty concerned with freedom. And we also have established big $100 million companies already playing in this sort of gray zone stuff. So I don't know what we do about that, but just my thoughts would be more team effort, just like what we're doing right now, more conversations and dialogue about these therapies and that includes patients, doctors, scientists, regulators, and the domain organizations that know each of those conditions very well. And it sounds like, based on the other presentations, early access to some of these therapies as we gather more data, maybe using third-party registries, and that's about it. So next slide. Thank you. I'll have the interview with JSRM out probably in a week or so, and I'll put it front and center on the website. So thanks for your time.
Thank you. Thank you for your time. And thank you to all our session 3 speakers for their important insights. And I also thank you for your flexibility and some of the shuffles that we ran into. I do want to try one more time to call on one speaker that we couldn't get a hold of. Bojing Kukosi Shabangu, are you still on, or are you available or online? Give it one more try. All right, I am not seeing any mics coming off mute, so we'll move into the panelist portion to see what questions or follow-up FDA panelists may have for our session 3 speakers. We'll start with Dr. Karumbayaram. If you can please come off mute and ask your question.
Good evening, everyone. This is Inshaq Saran. I am the CMC reviewer at the OTP. This question is for Dr. Bambi Kurli. I hope I didn't mispronounce it. Firstly, thank you for your wonderful presentation. And if I proposed to join BLA, under that part, who would you think would be responsible party for this BLA? And do you suggest that all the individual applicants apply one entity name, such as an applicant? What mechanism do you recommend to ensure consistent manufacturing standards, including supply chain management across multiple manufacturing facilities under the joint BLA?
It was a little hard to understand you, but I think you were asking how that would work across multiple institutions and sponsors to file a joint BLA. I think that's part of what we really need to push in on. But what we'd like to see come out of this, if this was at all possible, is something that looks like an availability of some sort of license for a product that could be made at multiple facilities, whether as part of this process, FDA would require that we harmonize the manufacturing across all sites, or whether it could remain that the products are manufactured in slightly different ways. I think the key part here is sort of pulling the data together across these different centers and saying, okay, so there was a slight variation in how it's manufactured at MD Anderson as compared to Baylor College of Medicine and other institutions across the country. But at the end of the day, the product is a viral-specific T cell that's targeting these viruses. Is there a way we can move towards having a license for that? Does that answer your question?
Yeah, that's why you couldn't hear me clearly. I hope people can hear me clearly now.
You're still sounding a little choppy, maybe somehow getting closer, but did your question get answered?
Yes, it got answered. Thanks.
Okay. All right, thank you. We'll move to Dr. Kokai. Did you have a question? Any questions for our speakers today?
Hi, yeah, can everyone hear me okay?
Yes.
Okay. So my name is Lauren Kokai. I'm a CMC reviewer in the Tissue Engineering Branch 2 of the Division of Cell Therapy 2. And my question is for Matthew Hewitt of Charles River, if he's still online.
Yep.
Okay, perfect. So in your presentation, you spoke about duplication, in aseptic processing simulation for cell therapy manufacturing. And you mentioned that thousands of doses are produced per year. So I was asking you to clarify if you're referring to a singular manufacturing facility that's demonstrated ample experience, or if you're speaking across all manufacturers, regardless of the experience of the individual manufacturer. And if you're speaking across manufacturers, could you please clarify the ASP simulation experience that is universal across facilities?
Yeah, maybe I'll address the last point first. I mean, by program, the APS is standard across the program itself because it's simulating the connection points for each individual process. And I think those connection points don't change by process from day to day. Because it's laid out within the batch production record that governs the manufacturing process for that product. And so I think this is something that could be implemented on a per site basis. But I think if we look at the testing that's required to essentially establish that we're working in an aseptic way for each process, whether it's APS or product, it's the same. And so the primary differences is one is using cells and media, potentially with serum, and the other is using generally something like agar. So, I guess what we are looking at is that if we are required as a CDMO, for example, to do 3 APS runs per program, and in some cases with some of the new guidance in Europe, it's actually 3 APSs per shift. And then we are in some cases in that guidance required to do qualifications of individual operators for what is termed critical steps, which are not well defined yet. Then if we have half a dozen different programs that are running at commercial, that would be over 100 APS runs per year if we were a 3-shift facility. And so the question is can we use the existing manufacturing data for the product itself as a risk-based approach to replace the APS? And remind that, for any product that we are producing or we are manufacturing, we work with our sponsors to submit yearly manufacturing updates to the FDA per program to establish the frequency at which we're running these processes. And I think if you look at the frequency that we're doing these processes, many cases we're doing tens, if not hundreds, and in some cases thousands per year. Those numbers are far exceeding the APS numbers that we'd be running. So actually, the more, if we look at this in a risk-based approach, the better data set would actually be the manufacturing data rather than the APS data.
Okay, I understood. That was very clear now. Thank you.
Yeah, of course. Thank you for the question.
Thank you. Dr. Tomar, do you have any question for our speakers?
Hi, my name is Sakshi Tomar. I am in the Office of Pharmacology Toxicology. My question is for Deborah Webster. You noted that the microenvironment in the murine and the immune factors therein are not directly translatable for assessing tumorigenicity risk in humans. Could you please clarify how you envision in silico and in vitro analysis would take these factors into consideration in predicting tumorigenicity risk in the clinical microenvironment?
Thank you for that question. I'm not an expert in that area. What I made those points because basically you're transplanting a human cell into a mouse, and cells respond to everything. So they respond to that environment, and we just feel like that's not a very relevant translatable model. In terms of assessment, it's like the in silico analysis would be looking at your cell line and making sure that the genome is stable, that there's no that looking at the genetic stability of the cell line as it's going through processing to the manufacturing and so forth. So again, I'm not an expert on the CMC aspect of it, but what I was really focusing on is that I feel like there has been a lot of experience that FDA has gained over the course of the first cell therapy to where we are now. And wouldn't it be possible to evaluate and consolidate that information, using AI to probe what has been conducted and what's been actually translatable into really showing an effect. And I that's why I mentioned the ELIScell example, because that was negative. And it did, the insertional analysis and so forth. And so even those things, did not really identify something that was seen clinically. And it may be that we may never be able to have a translatable tumorigenicity assessment for cell therapies. And I just feel like there might be a wealth of information within the studies that have been submitted by the industry to FDA for you to be able to interrogate that through AI. And I think what's really missing is I do believe that FDA has given guidance on the types of in silico analysis and so forth that need to be considered in a risk-based approach, but that they're sort of in various different guidances, and it would be great if they could be consolidated into a single guidance that considers really it from that whole risk-based approach as to what level of data you would need to be able to say, okay, it doesn't make sense to do an in vivo tumorigenicity study.
Thank you for that clarification.
We have a couple more questions. Circling back to Dr. Kurumbayara, if you have another question to ask.
Can you all hear me clearly now?
Oh, yes, much better.
Okay, thank you. Excuse me. This question is for Dr. Matthew Hewitt. So in your presentation, you mentioned eliminating aseptic process simulation for the manufacture of each autologous product. My question there is, what alternative controls do you propose to ensure safe manufacturing, given that the autologous manufacturing process may increase the risk of propagating pathogenic agents that could be present in the donor material?
No, I appreciate that. And maybe I would make a couple comments and clarifications there. One is that we would certainly want to continue to do APS for new products that are entering, going into a submission phase for an IND, because that'd be a standard part of moving into GMP as APS training runs and engineering runs. I guess the clarification there would be, is there a point at which, when we go into routine manufacturing, that we could move away from the APS studies, because the APS studies don't take into account the potential contamination from starting material. But what it does show initially is that you can perform the process in an aseptic manner. And I'm not sure that the starting material potentially being contaminated would necessarily constitute a risk that would be outside of what we would consider the normal operating procedures of manufacturing those types of products. And I would also point out that we treat all products within GMP as if they are contaminated, which informs the training processes behind a lot of the facilities. And so the initial APS studies would stress that as per the typical regulations around that. But again, I would come back to the point that the larger data set that we would be able to produce to show the agency that we are doing things in an aseptic manner would be the actual manufacturing process rather than an APS study. Hopefully that answered your question.
Got it. Thank you so much for the explanation.
Yeah, thank you.
And we have one more question from Dr. Kokai.
This question is for Jeremy Ewald. Still on. Okay, great. Yep, so you described the ASRM pathway, which enables physicians to outsource manufacturing to CFP or CDMO. And I wondered if the Common Contract Development and Manufacturing Organization enabled sharing of manufacturing information such that the manufacturer would be able to advise the physician on the best manufacturing practice for product types. And if this is the case, I was curious how products would move across pathways to scale. So if something showed clinical success in this pathway, how would it then move to mass manufacturing, and how would comparability be handled?
Yeah, that's a good question. And I know we dug a little bit into that in the interview with JSRM. I believe that the cellular processing facility has to have their own, they have unique requirements from the government in order to be able to make that product, for one. And then if they can show good data under the ASRM track, they can enter the PMD Act for that mass manufacturing. But I believe they have to start from square one. And also what's interesting is that PMD, that mass manufacturing one, if it gets that conditional time limited approval and then the endpoints are not met and it fails the trial and it's not going to be approved, they can still take that therapy and use it under ASRM on a case by case basis with physicians, and that happens sometimes. So yeah, it's complicated. I think that I think the regulations have been around for maybe about a decade, and still a lot of work to do.
Okay. Thank you so much, Melanie. That's the end of my questions.
Okay, thank you. There's any more questions from our panelists? Just giving one more sweep here. Otherwise, I think we're good. So I want to thank you to our FDA OTP panelists and to all of our session 3 speakers for sharing your insights and perspectives. This now concludes today's public listening meeting. I'm just waiting for the slide to come up for our closing slide. But on behalf of the FDA's Office of Therapeutic Products, I'd like to thank you for attending today's listening meeting, and a special thank you to all our public speakers for making the time to share your perspectives and insights on this important topic. I'd also like to thank the FDA OTP Working Group for their help with the planning of this meeting, as well as our AV support who helped ensure today's meeting ran smoothly. As a reminder, a recording of today's events will be posted on the FDA.gov website in the coming days. If you have additional comments to share, please add those to the docket, which will be open for public comment until October 17, 2025. To access the docket, type in FDA 2025 and 2018 in the search bar on regulations.gov, which is also in the meeting chat. In the months to follow this, following this meeting, FDA will develop a draft guidance on the subject of this meeting and post it on fda.gov for public comment. And thank you again for joining, and have a great day.